right, good morning everyone. Welcome to this last half day of presentation of Thesis from the report. As usual, we'll be doing three presentations and then taking a break and following up. I'm Olivier Gauthier, as you probably know by now. I'll be chairing and moderating this session. I have with me the members of the jury, Yas from Oviedo, Stefano from here in Ancona, Stefan from Sorbonne, and Max also from Sorbonne. Bernard is here with us. I don't know if he'll be partaking in the jury, not officially on my list, so we'll see. Yeah, everybody can ask questions. I remind you have 15 minutes to present your work. When you come to the 13-minute mark, I'll try to wait and get your attention just so that you know you have 2 minutes left to conclude. And if you see me standing up, it means you're grossly over time and you need to wrap it up immediately so that we have a bit of time for the discussion. So, good luck. Best of luck to all of you. And whenever you're ready,
Yes. Hello, everybody. Good morning. I'm Pei Yan, and today I'm going to defend my thesis on current difference of 4 intertidal macroalgae species to high temperature and low salinity stress under the supervision of Professor Lodge and PhD student Benedict. First, let's take a review on the global climate issues here. The marine heat wave has been classified in both in frequency and duration compared to the last century, as you can see, especially in the North Atlantic. And this impacts a lot on the marine life, especially the fundamental species like seagrass and macroalgae. When it comes to the marine heat wave, the species abundance in macroalgae loss, and there are some reports showing the local extinction in some southern habitats, and also the algae was replaced by some fast-growing species. By the end of this century, some common macroalgae growing in this area is projected to shift their distribution to the north. Also, especially this brown algae Ascophyllum nodosum may lose most of their habitats, and this fragmentation may make the habitats in a smaller size and also separated. In a regional scale, in the Baltic Sea North Sea continuum, as you can see in the graph, there is a very steep surface salinity gradient changing from the brackish water to complete salt water here. And in this area, the precipitation, ice melting, the river runoff brings great amounts of fresh water into this area, making this freshening continues the effects over decades. And specifically in this Strait of Skagerrak, China Marine Laboratory, where is my study area, also face the currents from the Baltic Sea directly, bringing the brackish water to this intertidal zone. And you can see this local log data showing that in green during the summertime, there are always some short period of time experiencing very low salinity and high temperature conditions in the shallow water areas. The extreme condition can be over 25 Celsius degrees and below 15 PSU in salinity. And on this Swedish west coast, there are 4 common canopy macroalgae growing in a vertical extension pattern here, as you can see in the picture, from the upper splash zone to the lower immersed zone. And in theory, there is an upper range that is constrained by, of this distribution is constrained by the physical stress of the algae they can tolerate. And in this graph, as you can see, the focus surrounding is on one of the 4 that is always immersed in water and seldom exposed to the dry air. So maybe there is a difference in the current. Considering the ecological service that can be provided by macroalgae, except just the foundation species that's stabilizing the structure community by providing foods and shelters for other organisms, there is also like carbon capture, nutrient cycling function that macroalgae can provide. So it is very important to understand the living situation of macroalgae on this intertidal zone. reacting to these summer extreme events. So here comes to my research questions, is that how these 4 macroalgae species respond to these simultaneous heating and freshening events in growth and also the photosynthetic performance. I hypothesize they have different response, especially with the change in the both gradients, meaning there may be an interaction with the temperature and salinity. Specifically, Fucus serratus may be the most sensitive one. And also, I hypothesize the photosynthetic efficiency is somehow correlates to the growth when they're being stressed, and there can be an early indicator. I did my sampling on the rocky shore near the Chanel laboratory, which I showed the location before, and I put each species one non-productive glaze into a bottle, culture bottle that filled with different salinity levels of seawater, and 2 replicates for each level. And then I put the bottles into a bigger water tank that can be heated by a current heater with control. I have 5 these kind of boxes that can create a gradient of temperature. And this is the actual setup in the climate room. And I conducted the stress experiments by heating and putting them into low salinity for 24 hours and followed by a recovery for 3 weeks under the condition of 22 PSU in salinity and 20 Celsius degree, just as the control. Before and after the experiments, I measured the area and the wet weight. I measured the photosynthetic capacity using pump, like taking the records in maximum content yield for several times. I also built the rapid light curve before and after the recovery. And also I tested the steady state relative ETR under the light condition. I conducted a PCA for all the variables and correlation, and I fixed the. I tried to fit the linear mix model for the growth response, and also did a selection after I used the best model to do the prediction. And here's the PCA results by species. As you can see, there are some autocorrelation in the one parameter. For example, the 4 FEFN measurements are autocorrelated together. And also the growth response variables are correlate positively to most of the photosynthetic parameters, except the alpha slope on the left. And by species, as you can see, the data points, the Fucus serratus have a very different distribution of the data points compared to the other 3. That may indicate a different response in them. The appearance and correlation shows here there are 2 parameters in the photosynthesis that are highly correlated to growth, but they have different changes over time. For example, the FEFM is more correlated when measured after recovery, which is the opposite for the EDR measurements. And by plotting them by species, and you can see they have different correlation, like either linear or non-linear here. And so you can see those samples in recovery showing a very low FVFM values and EDR values that may indicate a negative growth in area, meaning they are already damaged or degraded. And here is a bubble maps of the growth response in area and wet weight, which shows similar patterns here. Compared to the control, which is the data points at the bottom right, all other samples under different stress are decreased in growth. Especially in Fucus serratus, you can see there are many points in red and orange, meaning they already have negative growth, which they are damaged in parts. And you can look at vertically and horizontally compare the impacts created by salinity or temperature. And you can find the temperature has a larger impact than the salinity. But still, the Fucus serratus is already very stressed, even without the heating when it puts into a very hyposalinic environment here. And in the linear regression by species, as you can see, different response traits in the species. For example, the vesiculosis and the ascovilin nodism, they have similar response when the salinity goes lower, the heating impacts will aggravate a lot, but which is not the case in spiralis, which always keep similar slopes and the lines are grouped together. And for the serratus, also again here you can see in the red line, there's a very significant impact in the in the very extremely low salinity treatment, even with all the different levels of the temperature treatment. And for the selection of the mixed model, a linear mixed model, I picked this one with the three-way interaction in salinity, temperature, and species, and plus the random effect created by the culture bottles. And building the models by species, I found the spiralis and serratus did better in the additive model, and the other two did better in the interaction model. And looking into the details, I only find there are salinity terms and interaction terms are not significant in the vesiculosis model, but other than that, all are significant. And after that, I used this model I picked to build prediction in the surface response to see whether these four species react differently. As you can see, they are all showing the synergistic effect, which means the higher temperature and the lower salinity can have a strong impact on their growth. But for serratus, focus serratus, here you can see the contour line is almost straight, which means it has the simple additive effects on the response to the gradients. But in the other species, you can see the contour line are a bit curved to the upright here. That may indicate when the salinity gets higher, it somehow mitigates a bit the heating impacts. But here, specifically as covalent nodism, there are very good ability to grow in a hyposaline area here, which shows a still positive growth in a very low salinity. And compare my results to related papers. Also find there are similar threshold on the temperature that can make these microalgae species have dramatic drop in growth or even in the negative growth rates, about 26 Celsius degrees. And in the Fucus population in the Baltic Sea, there is also a similar synergistic effect on the high temperature and low salinic stress. But I noticed these findings always have longer stress period than my study. And for the photosynthetic parameters, there are also papers supporting that our continuous heating stress will make the FEFM values drops over time, and this is irreversible. And as you can see in the picture, they also found the visible damages and color changes on the blades when it's treated over 27 Celsius degrees. So, which also can be seen in my study results. And in the future projection of these areas, Skagerrak Strait, it is projected the current speeds will increase a bit, and also a strong inner flow circulations in this area, which means fresh water from the Baltic Sea may stay longer here. And also by the end of this century, the surface temperature may increase 1 to 2 Celsius degree in general. But for the summer and spring events, it could be much even worse. So that may be reached to the threshold that can stop the growth of the Fucus serratus first. But for the salinity dropped to 1 to 2 PSU, it will enhance the impact on the growth inhibition. And for the future study, I think it's important to know some chemical analysis of the compounds inside the species that may understand their self-defense mechanisms. For example, the ascophylin nodosin, they have the extracts can regulate the gene expression on regulating the osmotic pressure. And also, there are some intermediates and byproducts reported that can be toxic generated during the stress, but have continuous effects after stress. And to improve the experimental design, I think the field experiments or method experiments like the photo show here is better to simulate the heat wave events and also this summer extremes on sites would be better. And also extending the duration can understand more on the reproduction of this algae. And to wrap up, I did find different response in these four species, especially Fucus serratus being the most vulnerable here, and also showing the additive effects on the response to both gradients. And the other three are more showing a complicated interaction response to the temperature and salinity. And the drops in the photosynthetic parameters correlates with the low growth rate after stress. And to sum up, these short-term heating and freshening experiments already shows there can generate lasting slow growth and physical damages here. So this summer extreme events should be considered in the future forecast and conservation. Thank you very much for listening.
From 30 seconds over time, I think. I'm gonna turn over to the members of the jury. If you are a local host.
Just a question about your experimental
Yes.
design in a laboratory.
Yeah.
I'm curious, what kind of water do you use? I mean, the water is artificial seawater, or you use filtered seawater at the end?
In the laboratory, there is seawater tubes directly connected to the local bay.
Okay.
So it's I just use the local seawater and mix with fresh water to create the difference.
Tap water.
Yeah,
Okay.
yeah. So fresh water with the seawater.
And you don't know nothing about the nutrients.
Yes, for the nutrients, in my pre-trial already tested that whether they add nutrient or not, they react in the same bit because the nutrient is already sufficient for that in the seawater.
Okay.
Yeah.
Okay, so Sue first, then that. You have to say everything in these 15 minutes. I mean, for me, I'm not a specialist in this topic, but there is a lot of job, a lot of analysis, and a lot of information in the presentation. Just one advice: speak to people,
Yeah.
talk to people, I think that is better.
Yes.
Communicate better.
Yeah.
What you're saying.
Yeah.
And I have just two years, okay?
Yeah.
So the first one is you put in the first or second slide, you said that in Spain, you put Spain in green as one example of recovery after the Olympic wave of the 20% of aquatic. Can you tell me something about it? Because at least we can tell us the number 3 is Spain.
Number 3 is around UK, I guess, but this is for seagrass. Yeah. But in this paper, the researchers says there may be in this area a slight increase in the water temperature may have a positive effect on the growth for the species here, for seagrass species here. So, but it's not like a common case around the world.
Yeah, because I mean, Spain is very different. They're having an increase in the north area in
Yeah.
this case, disappearance of some species, retraction of the hair, for example. I mean, it's not so big. Just
Yeah, yeah. also curious about.
About you were talking about self-defense. Self-defense in some species. How is that? I mean, can you comment something about that?
Yes, here.
Yeah.
Because I found some papers that they use the extracts from the esculent medicine, but unfortunately I don't really know the the chemical names. Maybe it's a group of chemicals that can apply to the agricultural crops that they can find this improve the cell osmotic pressure regulation by affect some genes expression in other plant cells. So, yeah.
Are there some studies about transcriptomy or something in those species just to try to assess how changing the expression of some or the evolution of some methodologies there in those species? Do you know something about that? I mean, in those species that are under stress, if you know something about some transcriptomy studies to see how it's changing the concentration. I'm saying this because maybe it's connected with some mechanism that exists to face that stress due to the addictive wave and increasing of salinity in those species. Do you know something about that or read something about that?
Yes, I know because all these are brown algae I was working on. So I know there's compounds called ferrotonin, which the contents will change when it experiences low salinity stress. And this is also the compound that's related to the defense for the grazing. So they may be more vulnerable when they face the hyposaline and also the grazing at the same time. And yeah.
Okay. And then my final question is about you were talking about conservation effort. What is exactly your idea about what we can do for conservation? Because increasing in the magnetic wave is something that I don't know what we can do. What are exactly the meaning of conservation effort in those species that you were assessing?
In this,
It's your idea. It's exactly what we can do because we can do the conservation of those species.
I mean, like building building a natural reserve for sure. It like reduce the human impacts of this area, and also and also. Yeah, maybe.
Don't worry, Subaru. You got a job, I mean, a lot of data, your results.
Yes.
This is good,
Yeah,
well presented, so don't worry. I'm good.
Yeah, but it's
Thank you.
Yes, thank you very much for the presentation. I think there was a lot of information. So it's really understood the PCF, for example.
Yes.
There are so many
Yeah.
variables that were not really explained in the methodology, and the text is a little small, so I
Yeah.
cannot see what I was looking at. So,
Yeah.
first, could you say some word about the methodology? What does it measure to assess algal growth or not? What does it measure to assess go back to the methodology and briefly express again what was measured?
Yes.
I did not really understood the PCA.
Yes. Yeah. So these variables are from the growth in area and wet weight, and also the parameters here.
But which one
Yeah.
is then in the PCA?
Yes. Yeah. So this is a maximum content used by 4 times because I want to know whether they will change over time. So I measured them 4 times here, and also
You want to
Also,
What's the data table here?
what's the data?
What's the data table that you are running the PCA on? What do you have in
I have
as line and column?
I have all the growth here, West Wind and area, and also other photosynthesis parameters. And here is the data points that shows the treatments. Like the bigger the bigger in size is the higher treatment in salinity, and also the color. The red one is the highest temperature treatment, and the lower is the blue one is the 20 degrees. So here, basically, I wanted to compare by species whether they react to this response variable in a similar way or not. So the most important information is about this data point's location. Like they are more correlated to these photosynthetic parameters and growth here, or either they went to the other way. Like maybe there is very different pattern in response. So that's I will continue the next test.
So what I think that we should measure the change in weight in the method. What do you assess the change in weight for my body?
Change in weight.
Yeah, if you go back to the method, I did not see how you measure.
Yeah.
Here.
Yes. The weight is just weighed by scale. Yeah.
Okay, so apparently the two species have completely different way of responding to the change. Okay, then in your analysis you are showing figures where you have a linear change, you use the linear model, where it doesn't look like it's a linear change.
Yes, but this is like exploration for the trains that I want to see more clearly in here. It's not really clearly how they respond, and here is like a very initial idea to see how the slope changes in different salinity treatments.
But sometimes it looks like there is a kind of C effect where everything is fine and it doesn't come where it drops.
Yes, and that's I have the models here. That is the interaction model that shows there is like a response curve. It's not like a linear
But that's
effect.
also it's a linear model.
Sorry,
Is it a linear model?
linear.
Model. But what is the assumption behind this?
But this model include the interaction term of the two salinity and temperature changes. So which means the elevated temperatures treatment is also simultaneously affects how the level of the impacts from salinity. So it's like it's not like 1 + 1 = 2. It's like more than. So this shows it's like a curve here, as you can see from the prediction service model. So it's like there is like a slow lower salinity stress here may also enhance the impact from the heat. But this is not the case for the surroundings.
Thank you very much. Congratulations.
Thank you.
We'll move on to our next presentation. So, our next presentation is by Yana Tugai. So Yana, you have 15 minutes whenever you're ready. We'll start the time.
Good morning, everyone. Thank you all for being here. My name is Yara, and today I'm going to show you my thesis topic, which was on the effect of alexandrine chemicals on the theme of competing microalgae. So to start, just to give some background information about harmful algal blooms, the HABs, they are a natural phenomenon that happens in coastal waters characterized by rapid proliferation of toxic algae that can be due to biotic and abiotic factors such as light intensity and nutrient availability. Also, the intensity of the harmful algal blooms has been increasing throughout the years and it is likely that will continue. Out of the different species that are responsible for the HABs, Alexander minutum is an important one due to the harmful compounds that it produces, such as neurotoxins, but also the bioactive extracellular compounds, which I'm going to focus on due to the activities that they have. So they can have toxic activities against cells in a phytotoxic and homolytic way, against fish and shellfish in an ecotoxic way, and also against phytoplankton in an allelopathic way. So still just a little bit about the ecological consequences of the LL chemicals of Alexander minutum, just a small scheme showing that with the release of the LL chemicals, it can have negative impacts on fish, where they can accumulate these toxins and transfer them to the trophic web, but also on bivalves as well, and it can also affect the fisheries. But this project is focused on allelopathy. So first, I need to explain a little bit what it is. So allelopathy is described as a direct or an indirect effect that can be harmful or beneficial produced by one microorganism to another due to the release of toxic compounds, here mentioned as allele chemicals, into the environment. So these allele chemicals can confer benefits to the organism that produces them, and as it helps to the bloom formation, and also can inhibit its competitors. This inhibition can happen through the cell lysis, also inhibition of photosynthesis, and also to the production of reactive oxidant species. And all of that can lead to the allelopathic shuttle. So here we have another scheme where we show that with the release of the allele chemicals from Alexander minutum, they have the potential to kill the phytoplankton community, and then the phytoplankton community can release the dense organic matter, and these can then aggregate in the form of marine snow. This marine snow can then sink to the seafloor from the water column, and this is the allelopathic shuttle. It's the sinking of the marine snow from the water column to the seafloor. Allelopathic effects have already been studied, however, in only one diatom species, in Chaetoceros malariae, where it was observed that there was morphological modifications, also alterations in the photosynthetic efficiency, and there was also formation of aggregates. However, what happens to the rest of the phytoplankton community? So here the goal of this study was to see what will be the effects of the chemicals of Alexander minutum on other competing microalgae to try to understand the implications that the allelopathic shuttle can have on the phytoplankton community. So a little bit about the experimental design. First, we did an initial screening where we used 16 different microalgae, where we exposed them to the chemicals of Alexander for 2 hours, and we were looking at the morphology of the cells, there were cell lysis, and also for motility for the motile species. Then we did a bioassay, which was a more complete screening, where we did triplicates and also measurements through flow cytometry to assess red fluorescence, which is a proxy of photosynthesis, and also to see cell concentration to be able to do cell growth rate calculations. And then we did roller tank experiments. So here we have a little video of what is a roller tank. So the roller tank is this cylindrical structure that we put on top of this table. It is in constant rotation. And this constant rotation is aimed to mimic the natural conditions in the environment, so the water column, which then facilitates the formation of the aggregates also. So in this screening, we were using the cells of Alexandrium and of the allele chemicals, as using the cells there is a constant release of the allele chemicals in it would be, and if they would lead to aggregates, it would be easier to see in these conditions. So after that, of course, we wanted to see the chemicals themselves, so we went to a triplicate experiment with Photomonochelina, where we were using the chemicals of Alexandrium. So just a little bit about the setup. As I mentioned before, I was using 16 different microalgae and one species of Alexander minutum, one strain of Alexander minutum, so I had to culture them, and I also had to get the allele chemicals. To get the allele chemicals, I had to do a filtration with a 0.2 micrometer acetate cellulose filter of the culture. And then for the physiological measurements, to look at the cell lysis and motility morphology, I was doing microscopic observations. Then to have measurements on the particle size and complexity, I was looking at the forward and side scatter for flow cytometry, and also looking at fluorescence for the chlorophyll content and cell concentration for the cell growth calculations. Then for the robo tank experiments, to be able to characterize the size and their sinking speed, I was doing the analysis on image tray. So a little bit on the results that we got. These was the first results from the screening, and here is results in terms of morphology, more specifically on cell lysis, on Ligodinium polyhedra, and we have the control group and the exposed. And what we can see is that on the exposed group, we saw multiple cells that they showed an empty tetra, which is the outside structure of the algae, meaning that there was cell lysis here. While in the control group, we never observed it. This means already that within just 2 hours, the LL chemicals were able to reach to cell lysis and affect the pathology of these species. In terms of motility, here we have an example with Codomonas salina. I hope it's able to see. But we can see that in the control group, even after 2 hours, that the cells they kept their motility and were swimming randomly, while in the exposed group, there was no movement at all. That's any movement that will be seen was just like the continuation with the water flow. Meaning also that within 2 hours, it was also affecting the motility of these species. Then, looking at some of the bioassay results for photosynthetic activity, we have the red fluorescence, which once again is a proxy of photosynthesis, and then the different species that were tested on the bioassay. In blue, we have the control group, and in red, we have the exposed group. And what we were able to observe is that for most of the species that were tested, that after the 2 hours of exposure, that there was a lower value on the exposed group for the red fluorescence compared to the control, meaning that the low chemicals in just 2 hours got a reduction of the chlorophyll function or their content. Also looking at growth rate, we also observed that for most of the exposed groups that there was a negative growth rate, and meaning that the LL chemicals led to cell lysis and a decrease in the population. While for the control groups, most of them were showing positive growth rates, and even though there was also some negative growth rates, it was also less than for the exposed groups. Moving on to the roller tank screening, what we can see here is the different species that were tested, and then we were also looking at if on the control group we observed aggregates, if we observed aggregates also in the exposed group, and how much time it would take. So here we have, for example, Glycerazara, that after 4 days there was no aggregates in any of the groups, but then we have 4 other species that, for example, we observed aggregates in different amount of time only on the exposed group, and these aggregates were always very, very small. And then we saw that for Codomonas salina, within just 1 day, we were able to see aggregates both in the control and in the exposed group, and that they were also quite bigger. And here we already have some information just by the way that LL chemicals act, meaning that they can be having different intensity towards each microalgae that they are targeting, or also that each microalgae also has a different sensitivity to the LL chemicals. Here it's still with Codomonas salina. It was the one that we decided to continue working on, so it's the results of the first screening, so using only the cells. As I mentioned before, there are also aggregates observed in the control group. However, we can see that the aggregates observed in both of them are quite different in terms of color and in terms of quantity also, meaning that the LL chemicals do have an influence on the formation of these aggregates. So we moved to the roller tank experiments using only Codomonas salina, and decided just to show that we kept doing some physiological measurements. And then for the triplicates, well, here's just the 2 first experiments. We had 2 roller tanks per group. For the first experiment, we saw that for the control, there was no aggregates formed, while for the exposed, there was aggregates. In the second experiment, one of the controls showed very, very small aggregates, and it's very hard to see in the pictures and even in naked eye. And then, however, the exposed group was showing aggregates, and then on the last experiment, all of them were showing aggregates. What we can conclude from this is that, well, chemicals, they have an impact on the way that all of the exposed groups always showed aggregates. And if we go look at the control as well, on the ones that we formed, aggregations as well, that it was quite different, both in terms of size and in terms of quantity, meaning that the chemicals will have some influence on the aggregate formation. Then we also wanted to look at the size of the aggregates and the sinking speed. However, in this graph, we only have the results for the exposed groups and not for the controls, because as I mentioned before, the controls, they were very, very small, so it was very hard to follow them on image J. It was really impossible, so we were just looking at the exposed group, and we were trying to see if they were following the sinking models that say that the bigger the size of the aggregates, the faster it will sink. However, as you can see, for example, some of the aggregates that we saw that had the biggest size, they have the lowest sinking speed, meaning that our results are not exactly following the sinking models and indicating that there are other parameters involved on the sinking velocity. That can be the source, the porosity, and also the density of the aggregates. So, in conclusion, what we saw from this study that the other chemicals, they have a fast way of acting, as we saw in the screening and on the bioassay within just 2 hours. We already saw some differences in terms of chlorophyll content, so photosynthetic activity, and also in growth rate. And then it can also have some implication on the physiology of the microalgae, also leading to aggregate formation. We also saw that different species have the capacity, have different sensitivity to the allele chemicals, and also that the allele chemicals can influence on the aggregate size and the sinking speed. Also, these results are also relevant in terms of ecology and on biogeochemistry, as they have some implications. As we know, marine snow, they play a very important role in the vertical transport of the organic matter that provides microhabitats for zooplankton and also for microbial communities. So a change in these aggregates can lead to, for example, also change in the feeding behavior of zooplankton, and it can also change on the microbial community that are present in aggregates. And it can have also implications of, for example, degradation rates of the marine snow and less transport, for example. And thank you to everyone for listening to my presentation, but a special thank you to everyone from the lab that helped me with this experiment, especially to my supervisor that did a great job supervising. And I also want to thank the Institute for the funding and the lab for hosting. So thank you very much.
Thank you, you took 12 minutes and 30 seconds, but at the same time you were not running, I think you were in a good place. Yeah, then we'll thank you.
I was not running. Okay.
You spoke a bit faster, you were not running.
Okay.
We have ample time for discussion, so I don't know who wants to go first. Yes.
Congrats to everyone explaining some prerequisites and details. What are exactly those chemicals?
Those chemicals,
Have been purified, characterized?
no, they are not been characterized yet. But people are still, in this project, we were not characterizing the chemicals, we were just trying to look at the effects that we have, but they still have not been exactly characterized. Still,
But not in that species or not at all?
not in this species. We just know that they are.
Could be very interesting.
Yeah.
What is exactly?
Yes, it is, especially because we also see that they have different effects for different species, so it will also be interesting to, because maybe some of the microalgae species have some characteristics that are beneficial to them, that are not sensitive to these chemicals, and it's because of the composition that they have.
You started by the blood rules.
Yes.
Yeah. So, what do you think are the best, the main implications of your result for that so sensitive problem?
So here it's because like I started with the blooms because Alexandrium minutum is one of the algae that has been also occurring more often in the algal blooms, and it's the problem on the way of the phytoplankton. That what we wanted to see is that because when there is a bloom, is this massive proliferation of one microalgae species, and normally the algal blooms are studied in terms of the effect that it has on bivalves and fish, but not exactly in the phytoplankton community. So that was the goal. It was to try to see, okay, we have the bloom, we have the proliferation of one specific species, but what happens to the others? Are the others still able to cope with this proliferation? Are they not able to cope? And in the future also, like trying to see maybe if they will not cope, but maybe if they are able to come back. And,
Other, why are you not including statistics in your model graphs? Probably you did.
yeah, I did statistics, but I forgot to mention this.
Because you compare groups and you
Yes.
should put A, B, or something like this.
Yes, I know.
Because you have replicates.
Yes, I do have replicates, and I did do statistics, and yeah, but during
Do it.
yeah. And during the presentation, it's like I forgot to put it. Like we saw that some of them, we saw that some of them were significant, and the one that we saw like throughout the entire study and in the different parameters that we were studying that was more significant, it was always in part of the showing it because it's,
I guess almost all of them are significant.
yes, yeah.
It's good to present.
Yes, I know. When I was going through my presentation, I was like, yeah.
Okay, congrats.
Thank you.
Hello. Congratulations. So, right in the slide, I just curious, why do you use also affiliating criteria? I mean, this is a bad data.
Yeah.
And
We-
so I'm just curious why you also try identify.
Because we were trying, we wanted to try with some of the algae species that were available in the web culture, and that were able, and that was easy to grow, and that we just like that's like it was because it was like more the first screening that we just wanted to see because that's like it was only tested on microalgae species, and we just wanted to see like okay, is this also happening with other microalgae or not? And so we were just doing like a screening with most of the ones that we have. But even though it's a benthic species, that it is also important to study it on the way that like the aggregates can also lead to the bottom of the seafloor. So like that could also have some implications. Maybe it will not be like the most important algae to study out of all of them, but we just so just as a big screening.
Okay, good. And about the growth media, you said that you use the filtrate of Alexander, right?
Yeah.
And what is the growth media that you use? It's just
I used L1 and L1 plus silicates, depending on the.
it's a replaced growth media, right?
What, sorry?
With all the nutrients.
Yes.
Okay, because it will maybe later.
Yeah.
Or maybe you already done. You can also test the nutritional stress. I mean, when you have a nutritional several dinoflagellate roles, when you have a nutritional stress, they increase the toxin production, fights competitors. So maybe you can have also different results.
So-
Could be useful. I don't know if you.
So that is actually quite interesting, and it's like at some moment I thought I was over time or something. I thought that I didn't see you, so that's why I was also like wrapping up. But actually, so on the colonies experiments that we did, the second replicate, it was with a culture that was probably like with less amount of nutrients because it was turning green. So meaning that it was like not so healthy as well. And that's also why, for example, on the aggregates that we see that even on the control is more like with the green coloration than with the red one. And there's like, and here we were able to see that, for example, even on the red fluorescence, that for the control group, that even though it was higher than in the exposed, it was still like lower compared to the other two, for example. So it's like it was not on purpose that we used like this culture. It was supposed to be like a very healthy culture, like with the normal coloration, the reddish coloration. But here it was already like starting to get some nutrient depletion, and we're also trying to see for this. Okay. Thank you.
Yes, thank you very much for this very nice presentation. It was very qualitative, as I mentioned, there was not so many statistics. And so I would like to ask some question about the methodology.
Yeah.
So first, I think you already implied that the chemical compounds that you used in screening were extracted from
From
an example
Yes.
culture.
Yes.
So you don't know the concentration.
No, okay, so how we
And
do,
how realistic it is to develop
Yeah.
that kind of-
so what we do is that we would count by flow cytometry the cell concentration of the culture of Alexandrium. Then we would assume that it would be the same, that this concentration of the cells, it would be the same concentration for the allelochemicals. That's the way that we were doing in terms of calculating the concentration for the allelochemicals.
So in a nutshell, you say that you would find levels of concentration that you are using in your experiments, or you concentrated that azote?
Yeah. So for the first, so for the screening of bioassay and also for some of the robot tank screenings, we used a very high concentration to be just sure that we would see like some effect or not just be like, okay, it's because we used a small concentration. But then for the robot tank experiments with the photomonage, we used the concentration that is more similar to what happens in the natural environment.
How do you do that? Because the concentration, how do that?
Yeah, we were doing it like not with comparing with the LL chemicals because we did not count the LL chemicals itself, but saying that the concentration of the LL chemicals it will be the same as the concentration for the cells.
Okay, so in nature and or how would you measure those chemicals? Do you know something about that and how to measure that?
Actually, no, I don't know exactly, but
By chromatography or by any
yeah, there's like I don't know if it will be able, for example, also by flow cytometry to be used, but maybe not, maybe because maybe they will be too small, but maybe by cytometry that will be possible to do. Yeah, I don't think it would work as well. Yeah, but that is something to think about to be able
Yeah, I
to.
think
Yeah.
it's really to think about. Then, for the cell lysis and the mobility,
Yeah.
what criteria did you try to quantify or was it only a qualitative?
It was a qualitative.
So you did not try to count the empty cells versus.
No, we were not. No, we did not try to count.
Look at the speed of the mobility.
No, it was not that because that was not the point of the project, and it's like and since it was so many different microalgae that we were screening, we were just trying to see the overall effect. So we were not doing any quantification, just looking at it on a qualitative way. So, and what I was trying to see is like, okay, for example, in this case that I did not see any empty tepper on the control group, so that and then I saw quite a few on the exposed. So it was not in terms of numbers, but it was more of like none, a lot, some, because it's like qualitative.
But do you think it would be useful
For this project exactly, yeah, maybe, especially like in terms of cell lysis, to see like to see if it's able to, if it will decrease a huge amount of the population or not, but at the same time we can also have the results on the cell growth, which already tells us also like that there was cell lysis happening. And this is more to have like a visual effect and also here in the beginning we were not sure if we could use this algae or the flow cytometry or not, because maybe they would already like form big aggregates and would clog the machine. So it was more to just have an idea of if it's happening or not, and then to qualify it, we are doing by cell concentration.
So you don't think it would be useful to monitor the speed of mobility of cells?
Maybe, yeah, no, it could be, especially on like when we are putting them on the roller tank, because for example, because it's like Alexandrium is also a motile species, so it would also be important to see to try to see how fast they would encounter each other, because for the other species that are not motile, it depends only on Alexandrium for the encounter to happen, and well, on the rotation. But
Yeah,
here,
I think it should be
yeah, yeah.
Then also something you use a license as
Yes,
a proxy of family.
yes.
How is that? Because I'm not a specialist.
Um,
Why is vertical resonance a proxy of family?
Well, not as a proxy of primary production, but more as a proxy of chlorophyll function. If more as a, it's like
it was more as a proxy of like, okay, is photosynthesis like still happening or not, and more on the way of like of the health of the microalgae itself.
So it's a proxy of the mass
Yes.
of your clay.
Yes.
At a certain distance, so it's not.
Yeah. No. It's not. No. It's
The
not.
amount of clay.
Yeah.
is that.
Yeah. It was not production. It was more on the way to see.
And what was the weight?
What's right?
What is the wavelength?
Oh, I 450 to 480 nanometers. I think I'm not 100% sure. I know I have it on my report, but I'm not 100% sure right now.
So, I was very interested in this animal shuttle.
Yeah.
But in a simple word, just say what do you mean by that? What do you mean by the word shuttle?
The word shuttle is the sink from the column from the water column to the seafloor. That is a shuttle.
So when you actually sinking speed,
Yes.
why are they not sinking?
So probably it's because of porosity, for example, because, for example, with the aggregates that were observed with keto series, they were way more sticky and like less porous, while the ones that we observed here, they were very porous, so allowing a lot of water flow to come in between them. Also, that's like that would influence with their sinking speed, for example. That is what we think that could be the reason, like when comparing to keto series, mostly on the stickiness of the algae overall.
So you didn't do yet, you do that experiment for mass?
Yes.
Skeleton.
Mm-hmm.
Did you do such experiment with
Yeah, that is the plan also. The plan is to continue doing the screening because, as I was mentioning before, we first did 16 microalgae on the first screening, and then we started to reduce the amount of microalgae that we were using because of time. That was just like not possible to be able to arrive to the roller tank experiments with all chemicals. But the plan now it will be to continue the screening with both diatoms and dinoflagellates.
probably with this idea of there is no sugar?
Yeah.
That's going to that to be liter per day. It's less
Yeah.
1 liter per day. So
Yeah.
if they are not sinking, they will be maybe
Yeah, and it was also
well,
Yeah, and it was also what we were observing, like even on the road things, it was possible to observe that there was like not really a movement, that it was just like staying.
in the roller tank experiment, did you try to look at the size distribution of
Sorry?
In the roller tank experiment, you try to look at the size distribution of the aggregates in this file.
With time, no, we didn't look at it with time. We didn't because so at the end of 24 hours of the experiment, because the experiment would take only 24 hours, and then after that we would take the video analysis and photography to be able to use it on ImageJ, and we did not do any video or imaging during the time. However, we would be coming to the lab often to see if something was happening. And for example, even it was surprising. Because, for example, on the last experiment, I remember like the day before, when I left the lab, that I had like bigger aggregates, like the ones observed on the experiment 2, for example. But then on the next day, it's like they had already broken down. So probably that's like with the water flow, with the water flow in them that led them to the break. But it would also be interesting, and we're also talking about using a camera throughout the 24-hour experiment to be able to also see when does the aggregate start, both for the control and the exposed group, because we also observed that there was also difference in the timing, that the exposed would start aggregating in 1 hour, while for the control, if they would start aggregating, it was like after 6 hours, something like that. So to have exactly an idea, but also see like how they will develop throughout the time. For both of them.
Nature, how does it take for aggregation?
In-
Natural population.
That's what we do last question.
It's a simple word.
Yes.
Well, I guess it will depend on the species, but but I believe that it would also be like a small amount of time with all of the movements of the water flow that it would be that the algae race would form.
We don't know. Usually it's 4 or 5 days.
Okay.
Right. Thank you very much, Alan.
Thank you.
Hello everyone, my name is Tom and I'm happy to present today the results of my thesis research on the spatiotemporal fishing risk of large pelagic fish species in the Mediterranean Sea. I conducted this research at the Spanish Institute of Oceanography in Palma de Mallorca. First of all, what is the importance of large pelagic fish species? This group is generally characterized by their large-scale migrations. So, for example, the Atlantic bluefin tuna travels extensively throughout the Atlantic Ocean and into the Mediterranean Sea to spawn. Similarly, swordfish populates the Mediterranean Sea and has defined spawning areas in which they migrate to seasonally. Apart from this, these species are also apex predators in the pelagic food web, so they are at the top of the food web and feed on other smaller predatory fish, as well as micronectin, including cephalopods, for example. However, extensive fishing has often decimated these populations, which can have cascading top-down effects on the whole ecosystem. Talking a bit about the fisheries that target these species. It is estimated for 2022 that a record all-time high of 8.3 million tons of tuna and tuna-like species was caught. Additionally, tuna fisheries alone are worth $40 billion annually. Taking a closer look at the Mediterranean Sea, we have 3 species that are the most important commercially. This is the swordfish, the Atlantic bluefin tuna, and albacore tuna. These three species alone account for about 63% of all catches, and they are also hugely culturally important in the area as they have been exploited since around 11,000 years. The main gear types that are used in these fisheries are drifting longlines. Drifting longlines are deployed by a longline vessel, and it contains a line which contains then baited hooks. These longlines are left in the water to soak for a specific duration and are then hauled back in. They can also span up to 100 kilometers in some areas. Apart from this, we have tuna purseaners, and these vessels are mainly responsible for catching Atlantic bluefin tuna in the Mediterranean. They work as a more active gear type. They work by encircling, aggregating fish, and then capturing them in the encircled net. The question is then, how are we able to manage these fisheries sustainably? And we have different measures in place. One of them, for example, is through area-based conservation measures. So marine protected areas are one tool in which we can restrict the amount of fishing or the type of fishing that can take place. Apart from this, we also have marine spatial planning initiatives which aim to combine the rights of fishers with protected areas, also taking into account our need for renewable energies and, for example, offshore infrastructure, which is shown in purple here. Lastly, and most importantly in our case, directed fisheries management. And for highly migratory species, this is conducted by regional fisheries management organizations. In the Atlantic and the Mediterranean Sea, ICAT is the body that is responsible for the governance. They have numerous measures that are in place to manage the fishery, for example, through catch limits, through limits on the types of gear that are allowed to be used, on locational closures, seasonal closures, and also in general through effort controls. However, these things have one thing in common, and that is we need spatial data, either to be able to estimate it or to be able to enforce it later. There are different tools available to estimate where a vessel fishes, and traditionally mostly logbooks we use for this, which is the responsible of the skipper to note the amount of catches and the location for a given fishing event. However, they often come at a relatively low spatial resolution. In terms of satellite-based technologies, we have the vessel monitoring system, which sends the location of a vessel to a satellite every 1 to 2 hours in the European Union. However, this data is then gathered by governments and is not publicly made available generally. Lastly, we have the automatic identification system, which was originally developed as a safety tool. This data is publicly available, and it comes at a very high temporal resolution of up to 2 seconds, depending on how fast the vessel moves. This technology is also mandatory in the European Union since 2014 for all vessels above 15 meters in length. In our thesis, in my thesis, we tried to answer then where and when is fishing most intense, and do we see that spatial patterns have maybe shifted in the Mediterranean Sea during the last decade? What are in general the seasonal patterns of fishing, and do we potentially see changes in the season between years? And then lastly, to what extent are we able to capture the full scope of fishing activity using AAS-derived methods in the Mediterranean Sea? In order to answer these questions, we used AAS-derived fishing activity from Global Fishing Watch. And very briefly, Global Fishing Watch is a non-profit that gathers millions and millions of AIS data throughout the whole world and analyzes them based on deep learning methods. So Global Fishing Watch receives vessel positions, and then they recreate the track of a vessel, and based on the characteristic movement, they are able to infer whether the vessel is likely engaged in fishing or not. And this is a nice continuation of the talk we heard from Francesco Ferretti, because he was actually one of the people that developed this method. This data was then further filtered in R. We filtered it based on different parameters, for example, the distance to port, and information from regional fisheries management organizations' vessel registries. And then we conducted what is called an emerging hotspot analysis. And this analysis consists of two different statistical measures, the Gettys-RG statistic and the man-candle trend test. And what's important here is the Gettys-RG is a statistic that basically assesses how dense the clustering of, for example, high values is. But it is important that in our case, the data that we used is highly correlated in space, but also in time. So the cells that surround a given cell that we analyze are often also have a high value if our cell of interest has a high value. And in order to account for this, we group these cells together. So we analyze the sum of the cells at the time point we analyzed together with the same cells in the time point before, to then construct something that is called a space-time cube, where we have on the x and y axis our coordinates, and then as a third dimension we introduce time. The hotspots were then calculated based on these aggregated values, and we analyzed we calculated the man-candle trend test based on one location over time. And the man-candle trend test is it's a ranked test to analyze whether there's a linear trend within the data. Coming to the results, here we have the cumulative fishing hours over the last decade for both drifting longlines and tuna purseines. We analyzed in total 213 unique vessels, which together accounted for over 122,000 hours of fishing per year. However, one thing that is important is that we see basically distribution of fishing throughout the whole Mediterranean. We see areas of high activity, for example, in the Spanish Mediterranean coast, in the Adriatic, in the Central Mediterranean Sea, and off of Cyprus. One thing that is noteworthy also here is that we do not appear to have fishing activity in the southern Mediterranean countries. And this might be due to a lack of adoption of the AAS system. So these countries do not employ or do not have mandates for using AAS, and thus we are not able to infer any activity along these areas. Taking a closer look at the longline hotspots, we have identified longline hotspots with longline vessels from Italy, Spain, Cyprus, Malta, and Greece. And here we have shown the percentage in which a given cell was considered a hotspot in our analysis. And we see that we have persistent hotspot areas in multiple areas in the Mediterranean. For example, in the Spanish Mediterranean coast, south of Sardinia, in the Central Mediterranean, and north of Cyprus. We then try to match these areas with known ecologically important sites. So we know that in some areas, Atlantic bluefin tuna and swordfish gather to spawn, and we try to overlap this with our high visual fishing activity. So we see, for example, of the Spanish Mediterranean coast, this is a known spawning area, particularly around the Balearics for Atlantic bluefin tuna, as well as for swordfish moving more towards the Strait of Gibraltar. Additionally, also the Central Mediterranean and the Tyrrhenian Sea is a known spawning area for both of these species. Then we calculated our trends over time, and what we see here is that there are no general patterns. So within each main area that we identified, we do see some small-scale changes. However, the broad areas do stay consistently the same. And I would like to highlight one area here in the south of Malta, because this area saw a lot of high fishing activity, and we know from previous research that this area is also frequented by a lot of sea turtles, and there's a lot of bycatch rates in this fishery particularly. We also see an increasing trend in fishing activity, so this tool can help in identifying these areas and then potentially deploying targeted observers that that record information on bycatch to further assess this. Taking a closer look at the annual difference between the fishing activity, again, supporting what I said earlier is that we see that the general areas stay consistent between years. Regarding the longline seasonality, we see that activity of longliners usually starts in spring, reaches its peak during summer, and then falls off again in fall, and we have virtually no activity during winter. And this matches quite well what we know about the species migrations and when they are most abundant in certain areas. Regarding the purseine vessels, we've identified purseine vessels from Italy, Croatia, France, Spain, Algeria, and Tunisia. And we identified areas where they are most persistent around the Balearic Islands, in the Mediterranean Sea, close to the Italian coast, and in the Adriatic. We were then able to link two of these areas to known spawning grounds of Atlantic bluefin tuna, which is again the main species targeted by this gear type. Here I would like to focus your attention on the trends around the Balearic Islands, because they show an interesting pattern of a decrease towards the south and then an increase towards more of the north. And this is also supported by looking at the annual distribution of the effort in this area. So we see that it shifts from the south to more northern areas and into the Mallorca Channel. And we know from previous research that the distribution of tuna when they spawn is dependent on a dynamic oceanic front. So here is a frontal region that moves between years. And we hypothesized that we were able to show the movement of this front through using fishing activity data. Similar to longlines, purseine also have a very distinct seasonality, dictated also by the regulations of ICCAT, in which the fishing season usually lasts from May to June. And we see that it peaks during spring and then falls off during fall, summer, with basically no activity during winter. However, if we see persistent activity during summer and fall, it usually falls within this area, so the Adriatic and the Central Mediterranean. And this might be explained by the fact that Italy and Croatia are the only two countries that are allowed to fish smaller juvenile bluefin tuna, which stay in this area all year round. And they use them then for fattening farms. There are, however, some technical caveats that persist with using AIS-derived fishing activity. For example, captains are able to turn the system off. We also saw some coverage changes over time. So with the acquisition of more satellites, more terrestrial receivers, we also received more data which could mask real differences in fishing activity. And in general, we identified a bias towards larger vessels over 15 meters and vessels that are flagged to European countries, reflected by the fact that we do not see activity in the southern Mediterranean coastal areas. In general, these results are important because they can be indicators of changes in the distribution of the target species. And this is especially relevant with climate change, because a lot of species are expected to redistribute their habitat with warming temperatures, and using fishing-derived positions, we might be able to follow these species distribution patterns. Apart from that, we were able to also follow a dynamic oceanic front and its position around the Balearic Islands. They're also important to be able to assess the bycatch risk that, for example, sea turtles have in some areas. Other than sea turtles, there's also large risk, for example, for pelagic sharks or other unintended captures. And in general, these results emphasize the utility of using AIS-derived fishing activity due to its fine-scale spatial resolution. In contrast to this, ICCAT, for example, gathers reported information on effort and catches from each contracting party in the area. However, this is often at a quite coarse resolution, which does not allow detailed analysis, for example, of bycatch risk or of other interferences with the habitat or the whole ecosystem. To conclude, we conducted the first Mediterranean-wide spatiotemporal assessments of both drifting longliners and purseiners. We were able to then link this activity in some areas to known ecological features such as spawning grounds, and we identified in general broadly consistent spatial patterns within each major hotspot. However, we also saw some small-scale changes, which might be explained by changes in distribution of the species or other oceanic features. In general, this underlines the importance of vessel tracking, for example, for fisheries management. And some other studies have already started incorporating AIS-derived fishing into, for example, catch per unit effort metrics, which are an important metric used in stock assessments, for example. Apart from this data can also aid in analyzing spatial or seasonal closures, in which we know that a particular species is most vulnerable, for example, during juvenile stages. We also identified some future approaches, and we see a lot of value in combining this data with other data. So, for example, combining it with logbook data or catch data can give more detailed information, for example, for the use in catch per unit effort metrics. And also, there's a lot of utility in overlaying habitat suitability models with this high fishing activity to be able to estimate how much at risk a given species is at a given season, for example, at a given time point, which is necessary information for managers to be able to, for example, conduct seasonal closures or spatial closures. And with this, I would like to thank you for your attention. Again, thanks to my supervisors. I really enjoyed your supervision, and I had a nice time doing my thesis. Thank you.
Nearly perfect use of your time. Dr. Bars?
Okay, thank you very much. Yes, it was very dynamic. Really, maybe too many examples of the different situations. So, what I would like to ask you is, how would you try, if you had a 1 more month, 2 more months, to confirm your hypothesis of the change in the frontal, the circulation and position of the front? What kind of data do you need and how would you proceed?
The frontal region is mainly driven by the fact that less saline Atlantic water comes in this area and then meets with more saline Mediterranean water that has been longer in the Mediterranean. So then using, for example, salinity parameters, you should be able to follow this frontal and using other oceanographic information.
Which, such as?
Salinity.
What data are already available that use the same framework of data analysis?
Copernicus has a lot of information on this. They have different models on, for example, the sea surface temperature, the salinity. Chlorophyll A could also be something that we could look at, which is also important for them because the Atlantic bluefin tuna spawning is related in parts to also continental areas because they put their larvae in areas that are not too productive, but also that are not least productive. Because the theory is that in areas with very high production, we also have higher predation rates. They try to find a middle ground between this.
Okay. And what do you look when there are already people physical working on the change in circulation? Do you dig into the literature?
Well, the main
To support your hypothesis?
Yeah. The main paper was conducted by another researcher working at the institute I was at, so they also have a big physics department. And the novelty in their approach was that they were able to show that the patterns are moving off this front. This paper was, however, published in 2014, and I do not think that after then there were approaches to quantify the movement of this again.
Yeah, so maybe another question. Yes. Why did you aggregate the pixels around the position of the body? I don't get it. What was the reason?
Around the
When your data analysis schematic that you showed, we have the pixels where the code.
yeah.
And it looks like you were aggregating
Okay.
the pixels around.
Because in this sort of spatial data, the problem is often that we have a lot of spatial autocorrelation, not only spatial but also temporal. So if a given cell has a high value, it is also very likely that the cells surrounding it have a very high value. And in order to account for this, we aggregated them together. Because then we reduce the influence that this has on the overall analysis. Because in general, this data is also very, very highly skewed. So we have a lot of fishing activity where we record 0.1 hours in a given cell. However, what's important is that we analyze areas where there's a lot of high activity. So this is why we aggregated it in this way. And for example, it would be prone to outliers. So for example, if we have a cell that lays right here, which for some reason has very high activity, our statistical method will not be able to catch that. However, we argue that in our case, and based on the distribution of in general fishing effort, this is very rare to occur, because we also chose a resolution of 0.1 degrees by 0.1 degrees, which for this sort of analysis is a very fine scale resolution already.
Thank you, Lars. Excellent. Congrats. Very good presentation. I have just some figures. The first one is, what do you think about the real impact of the fisheries that is not controlled by ICE?
It's a very good question.
In the data that we have today, because can you have any idea about what is the real impact of that data?
It's hard to quantify the impact because, especially like nowadays, we have, apart from AIS, there's some other data sets that are being used for this. For example, satellite-based images, in which previous research was able to show that we might miss around 50% of the whole activity in the Mediterranean. However, this does not differentiate between gear types. So the majority of activity is through bottom trawling, which might make the figure a bit lower if we look at longliners and purseaners. And the problem with satellite-based images is that we do not know the gear type. And since my task was to assess these specific gear types targeting large pelagics, it was difficult to incorporate different data. But in general, we estimate, or it's estimated that in some areas in the Mediterranean, it can be up to 60% that is not publicly captured through AIS.
More, maybe something that I just missed, but how, something technical, how is the evaluation of the accuracy of your models? I mean, because maybe I'm just trying to think how to control that the results you are presenting here are real. I mean, the accuracy. Is there a way to control that this is the level of confidence? I don't know.
So in
No,
the
you never talk about that. I mean, I don't saw this data is also crazy. And to trust on what you are presenting, but how to control the accuracy of the results?
So in the original publication of the method, which was in 2018 already, they had numerous approaches to confirm what they were seeing based on the AIS data. So for example, they compared it with effort information that's available from regional fisheries management organizations, which correlated quite well with the hours, not perfectly, but I think it was an hour value of 0.6, so it correlated quite well. And apart from this, they then also always set aside a training data set in the deep learning methods with a confirmation data set. So they had generally quite high, it was quite high, the assessment of how well the method captures it, which it changed depending on the gear type, but I think for most gear types it was above 88%, where the fishing activity was correctly assigned using this method.
And this is very just how do you feel as a marine biologist about advance of the deep learning philosophy the changing the role of the marine biologist in front of all these new way to infer data that in the past you have to know and you have to be in vessel and use observers to have real data because all the data was not accurate. The question is on that because you were working on that, so maybe
Yes.
what do you feel? I mean, okay, I have all the data, I am using this script, these are the results. What?
I think it's important that we, on the one hand, we acknowledge the potential of using this sort of data because it's available in vast amounts of numbers, and also the number of publications that use AIS to estimate fishes have like skyrocketed the last years. So there's a lot of potential in using AIS data, using other big data sets that we have available. Because the advantage is also that it's less expensive. We don't need to send anyone out to get it. But it also comes, of course, at the risk of overly relying on these methods, using them without fully understanding the limitations. For example, if you have a bias in the number of vessels that use them, in the countries that use them, it's not going to be a perfect representation of fishing. And I think this is especially important to keep in mind, not as okay, we don't need observers anymore, we have this technology. It's not like this, because we still need, the data observers get is impossible to get in any other way. So I think we should see this as a supplement, as a very powerful supplement to the methods that we already have, and also to potentially better target where we put observers, because with this information we know where the fleets are going, we know where potentially we might apply catch areas, so we can target observers and make it more cost efficient to send them on these boats.
It would be very interesting to connect those results, those data with the land data.
Yeah.
Because as my experience, most of the land data is not.
And we know also the previous research has combined AIS with landings and logbook data to also assess the amount of misreporting that happens. However, I think there's also a lot of fishermen, not in all countries, that are really trying to follow regulations. I think this is also important to mention, that do everything they can to follow a lot of regulations that, for example, ICANN has put in place. And it's also not fair to blame everyone. But of course, we have to a large degree also misreporting, underreporting, especially in the Mediterranean Sea, which is heavily exploited. So, thanks.
Yes. Just want to ask you, what is the take-home message from your study for future?
It's difficult to give an overall, but I think this information is particularly valuable for ICAT in this case, because showing ICAT, especially in comparison with the effort data they receive from countries, so it's not necessarily ICAT, but it's the countries that send the data to ICAT, and then ICAT tries their best to make sense of what they resend and trying to disentangle this. However, if we are able to show what this technology can show, what vessels, we can actually bring it down to a vessel level. We can see which vessel went where at what time. I think it provides a lot of value for regional fisheries management organizations to assess where there's high activity, where do we have vulnerable areas. For example, with swordfish, we know that between different life stages, they are differently vulnerable. So the juveniles also occur mostly around the coast, whereas adults move more towards offshore waters. And knowing this, we could maybe potentially target spatial closures along with seasonal closures when vulnerable juveniles are targeted.
Great. Thank you very much. Congratulations.
Thank you.
Congratulations to the three of you, and we're taking a 30-minute break. Coffee break as usual.
Okay, it is just 11. Welcome everyone to this last session of the presentation for the master thesis. Last but not least, we will have today three for sure excellent presentations. Just to provide them. I'm Jackson Correto from the University of Oviedo. I'm going to be chairing this session, but I will have a great company with Camila Roberta here from Ancona, Gonzalo Machado from the University of Oviedo, and Nico Weber from the University of Oviedo. Okay, remember that the three of you will have 15 minutes for your presentation, 2 minutes later I will stand up just to let you know that you are on time. Joe, the floor is yours. Go ahead.
Hello everyone. My name is Sylvia and I'm going to be presenting functional originality and risk in fishes of the Western Mediterranean Sea and how these can be novel perspectives on conservation prioritization. I did my thesis at the Oceanographic Center in the Balearic Islands, and I was working on the Western Mediterranean Sea, which you can see here. So we are here right now, we're quite close. The Western Mediterranean Sea is the part of the Mediterranean Sea that spans from the Strait of Gibraltar in the west to the Strait of Sicily in the east. It is a global biodiversity hotspot, has, for example, many endemic species, but it's also a threat to several human impacts, such as overfishing or intense fishing pressure and climate change. So we need marine conservation in order to preserve the ecosystems and their function. And as always in conservation and in politics, we have limited time and resources which we need to distribute over species and areas. So we need to know where to start with conservation. And there are different perspectives on that. I'm going to be explaining or focusing on two today. First of all, we can look at importance. So for example, we might want to preserve an important habitat or an area that has very high biodiversity. We may also want to focus on keystone species that are very important for the ecosystem and or the habitats that they occur in. And then on the other hand, we might focus on urgency. So on species that have very high extinction risk and again, the areas that they occur in. And very often, these two perspectives are combined, which is what I'm focusing on. So I'm working on species level indicators of functional originality and different aspects of risk. What is functional originality? It's an aspect of functional diversity, and functionally original species are such that have very distinct functional characteristics, and those are often measured as traits such as body size, body shape, or fecundity, anything that relates to the species' fitness and performance. Functionally original species are very important for the ecosystem function. They're disproportionately supported, and they're also important for ecosystem resilience. So it is interesting and important to identify them and protect them, which is, however, not often done yet. So it's a bit understudied, and that is where my study comes in. So I focused or aimed to identify the most functionally original and at-risk species among fishes of the Western Mediterranean shelf and slope areas. And then working from that, I compared the conservation priorities across different indicators, and I also investigated into spatial patterns in the abundance of the species highlighted by my indicators. In order to do so, I needed three types of data. First of all, we need survey data to know the abundance and distribution of fish species in the area. Secondly, we need functional trait data, so ecological information on the species. And lastly, we need risk metrics. I'm going to be going in detail a bit more on all of these three data types now. So for survey data, we looked at the International Bottom Trawl Survey in the Mediterranean, short MIDITS, and we looked at data from 1999 to 2021. In total, about 13,000 trawls in either the shelf area, so the purple dots here, I hope you can see that, anything up to 200 meters depth, or the slope areas from 200 to 800 meters, and green dots in this graph. We analyzed the two separately because the species communities are known to be quite different in these depth strata. We looked at Tilossi and Chondrichthyes, so bony and cartilaginous fish, that occurred in a minimum of 1% of the hulls that we analyzed, and we looked at abundance data as opposed to biomass, so number of individuals per trawled area. In total, we analyzed 176 species. Now, for functional trait data, I relied on data that has mostly been obtained within the Be Useful project, with which I'm affiliated, and we based our analysis on 17 categoric or numeric traits that describe the morphology, the behavior in feeding, the life history and reproduction, or the distribution of the species. And lastly, for risk metrics, we looked at 4 different aspects of species level risk. So first of all, we included the IUCN Red List of Threatened Species categories that measure extinction risk based on demographic parameters such as population trends, and categorized them in 5 categories ranging from least concerned to critically endangered. Secondly, we looked at trait-based indicators of sensitivity to fishing pressure or climate change, so 2 of the most prevalent pressures in the area. And lastly, we included taxonomic scarcity, so just the inverse of the relative abundance in median survey data of the last 3 years of the study period. Now, how do we measure functional originality? There's a lot of measurements that have been proposed for this. All of them rely on functional trait data, and I'm going to be, well, I used 2 of them. From the functional trait data, what you always do is you want to have species pairwise dissimilarities. So you use a distance measure, in this case the Gowers distance, to show how dissimilar species are, and then from that resulting distance matrix you can calculate the first originality metric, which is functional uniqueness, and it describes the mean distance between the species and its 5 nearest functional neighbors. So a species that has high functional uniqueness is located solitarily in the trait space, if you will. And then secondly, for the second originality metric, I used a principal coordinate analysis on the distance matrix and calculated functional specialization as the Euclidean distance between the species coordinates and the functional space centroid, so species that have high functional specialization are located further away from the trait average of the community. Following the approach that has been proposed by Pimienta in 2020, that's called fused indicators, functional uniqueness, specialization, and endangerment, I then combined the functional originality metrics with a measure of endangerment. In the original indicators, this is the IUCN categories, and we extended this framework by the 4 different risk metrics that I just explained. So the result are 4 different risk-weighted indicators, and based on these, we then were able to rank our species according to the different indicators, and for interpretation, we focused on the top 10% within each indicator. For spatial comparison, we also looked at the abundance-weighted mean indicator value within each indicator, and we did that for 2 spatial scales. So first of all, for 0.1 degree hexagonal grid, that looks like this in a close-up. So we just pooled hulls into grid cells. And then secondly, the GCFM geographical sub-areas, which are commonly used for management in the area. I hope you can see it. That's they're just a bit larger scaled and very often referred to in the Mediterranean Sea. Now, moving to our results. As I said, we analyzed shelf and slope separately, so these are the top 10 species in the 4 different indicators for the shelf community, which consisted of 118 species, and 33 of them were top species in 1 or multiple risk-weighted indicators. So that already points to 2 important messages. First of all, a lot of them differed, which indicates that it's important to use different indicators and facets of risk, because they actually did differ quite a lot. But then also 12 species had a top score in more than 1 indicator, so that is, first of all, points to their conservation need and shows how these different pressures and risk measures are also related to some extent. And then what you can also see, and it's quite to be expected with the literature, is that Chondrichthyes, so sharks, rays, and slates, were quite highly represented in these top species. They made up 1/3 of the data compared to only 14% in the species pool. and they also had significantly higher values in all indicators. So to sum up, they're more functionally original and at risk according to these perspectives. And in the slow community, the picture was quite similar. Again, from 106 species, 29 top species, 16 of them were in multiple indicators, and chondrichthyes were highly represented, even a bit higher than in the shelf. And what's also notable is that the shelf and slow communities were indeed quite different, as we expected, and also the top species were different. I think 8 species were in both top species groups. Now, how does this relate to conservation priorities? The top species should all receive more conservation attention because the different risk measures measure distinct facets. Then at the same time, if you want to funnel this a bit down, then the species with high ranks on multiple indicators are of particular interest. So here I just noted down the ones that had high ranks in 3 indicators in the shelf community, and then in the slow community. And among those are particularly noteworthy that are currently not protected or insufficiently protected or not prioritized. So for example, the common torpedo is an elasmobranch, which are generally focused in conservation compared to teleost fish, but it still has a red list status of least concern in the Mediterranean Sea. So that could be an indicator, or that could be a result to look at more in our data because it has high sensitivity to fishing pressure and climate change, as well as quite high rarity and functional originality. I also would like to highlight that it is still important to look at the species that are not necessarily at risk yet, but have high functional originality. I'm not discussing this here in depth, but it was part of my thesis. And so, for example, the transparent goby, Apia minuta, is very functional original and not currently a conservation priority, but it would be interesting, especially because this topic is quite novel in the area, to go into this a bit more in future studies and see whether these species should deserve more conservation attention. Now, with regard to spatial conservation priorities or spatial comparison, I'm here presenting the maps for functional originality and sensitivity to climate change in the top row and fishing pressure in the bottom row. And then, as I said before, we're looking at community mean values in the left column and the abundance of the top 10 species in the right column. I hope you can see this. I was hoping for a bigger screen. But something that came up in all comparisons was that the variability was quite fine-scaled. So sometimes we have cells with extremely high abundances next to cells with low abundances. This is why I also decided to only show the fine-scale comparison here, as opposed to the larger scale comparison, because this high variability masks a bit the patterns also across GSAs. Now, the trends that we can see in sensitivity is, first of all, we have quite low abundances and mean values in the eastern part of the study area, so here. And this could potentially be a bit of an east to west trend, while we have highest values around the Balearic Islands, around the Alboran Sea, which has been highlighted in many studies before. It is an area that is influenced already by the Atlantic inflow, so that's why it's a bit different environmentally. And then lastly, the Sardinian shelves and slopes had high values as well. One possible explanation would be that Sardinia, or like generally the islands, experienced a bit less fishing pressure by bottom trawling, which is the most impact. So that could explain why the sensitive species gathered there rather than on the mainland shelf. Interestingly, the patterns in functional originality and scarcity or endangerment were a bit different. So here we don't see this eastern low abundances, but we still have high values in Sardinia and in the Alboran Sea. So these areas are definitely highlighted, as have previous studies found that looked at different biodiversity facets and functional diversity facets. Now, one thing that is very important to note about functional originality is that it is comparative. So here it's comparative to the studied species pool. Unfortunately, we weren't able to include the scarcest fish species. If you remember, we had that filter of 1% of hauls. So that would be, of course, very interesting to include in further studies, because these are potentially the most functionally and at-risk species. Then functional originality and rarity are also scale sensitive, so if we were to do this analysis on a smaller scale, for instance in just one habitat type, probably the results would be quite different because the species communities work differently. And then lastly, the metadata is conducted for demersal fisheries, so here obviously they are very highly represented and a lot of pelagic species aren't within the species pool. Then what's also quite important is that functional originality metrics are sensitive to methodological choices. Especially here we tested the sensitivity to input trait data, and that's also something I'd like to work on in the future, as when you take out single traits, the rankings do differ, and that is of course something that should be looked at in the future as well. And then, of course, we couldn't look at all aspects of sensitivity and rarity. They are quite complicated, especially the consequences of climate change, so that'd be something to also work on in the future. Now, some take-home messages. I hope I could convince you that functional originality is important. It should be considered in biodiversity research and conservation planning in the Western Mediterranean Sea specifically. And we also showed that a set of indicators, especially of risk, better captures the different important aspects of conservation need. Our analysis highlighted, as previous studies, that convictus are specifically endangered and at risk. But also we showed that single tedious species shouldn't be neglected, and we demonstrated the importance of the Alboran Sea, the Mediterranean islands, as spatial hotspots. Thanks for your attention, and to my supervisors and colleagues for all their support.
Thank you so much, Rosa, for the nice presentation. So now we have time for questions. So, you want to start with it?
Sure. Thank you. It was very, very nice. The initial presentation of everything was really good. I have a few questions and curiosity. The first one, since we are currently working also on functional traits, because we are involved in this project led by one of, actually was, she is an alumni from IPRC. She's a PhD student here in our lab. She's collecting functional traits for coral-indigenous organisms. So I know how difficult is this topic. Also because there is a lack of information on many species. I think that you can add an additional limitation that is depending on which functional traits you consider. Because, for example, life cycles, habitats where they may add eggs that can be different also from where the species lives. So, I was looking at the maps, and there are no data on the southern Mediterranean, so on the coast along the African continent, because
survey doesn't cover it.
We have the similar problem in one publication. And also, you consider the only fish, you know? But you know that in most of the cases, fish are related to specific habitats, no? And these specific habitats are mostly built by plant colonies. So do you think that adding the similar processing of functional traits on maybe not considering all these fish species are limited number and linking their functional traits with the habitat in which they can have in more tailored conservation measures?
Absolutely. Yeah, for sure. I think you're highlighting an important point here, which is how do we relate functional originality to ecosystem function? And that, as you say correctly, is completely dependent on the traits, which is why I decided to only do this on fish for now. So the metadata does also include invertebrates. like cephalopods, for instance, but also the trait-environment relationships and the the functional roles that are described by a certain trait differ in species groups. I mean, this is also true within fish, of course, but that is exactly why I decided to do it for this now. But you're absolutely right that the other species groups should be looked at. And then something else that plays into the selection of traits, as you say, is the data availability, which is usually, for example, a lot higher for commercial fish species. That's exactly why you couldn't include the rare ones right here. They would be super interesting because functional originality is also to some extent correlated to rarity. So yeah, and maybe one last point to the traits is that here I think what we could very well picture is dystrophic interactions. So we are quite, these analyses show quite well the distinction between top predators, small pelagic fish, which are super important in the med. But for example, other roles of fish in the ecosystem, like ecosystem engineering, which exists less in fish, but it does exist, is a bit underrepresented here because it hasn't been studied and it's really hard to find trait data for 180 species. So yeah.
Just another curiosity, also, consider the data on courses. So in the high season, you decided not to have this data or just not available?
The MIDITS data.
In the middle of the Western Mediterranean, for example.
Absolutely, these are super important areas. Of course, they are understudied, and the MIDITS data focuses on the shelf and slope, I believe, up to 950 meters. But could be wrong. So yeah, we just focus on the shelf and slope areas. They are really important ecologically, but the deep seas are definitely understudied and yeah, important.
Yeah.
Well, Sylvia, congratulations. This is really amazing work. I think showing here the amount of work that you have developed in this is reflected here in data. You think what you describe. A very nice picture, very nice diagram, good combinations of both. Some of the maps, you are right, that were a little bit small. Yeah, you can play the screen. You're going to kill me back. Can you just show me, please, again, and we can go to it, this slide where you explain the method to complete the functional originality?
Yeah.
Can we go to that one?
Of course.
Because they really want to understand.
Here?
Yeah. So you've got a matrix of the species and traits. From there, you can make another matrix between different species. Okay, can you develop more, please?
Of course. Yeah, so we have the species in the original data set. We have all our species. The species list is derived from the MIDITS survey. So all the species that actually occur in the area. And then we have all the traits that we're analyzing in the columns. And then what we want to do is we compare species. So we create a distance matrix that in each cell has the comparison between all possible pairs of species. There's different distance metrics that you can use. The Gauss distance is the most used because it can combine categoric and numeric data. And it also allows for weighing of the traits, which can help. We didn't do this with like an intention, but we made sure that all the traits contributed equally to the dissimilarities, which is sometimes a problem when using categoric traits. And then from that distance matrix, yeah, you isolate the species that have the highest dissimilarity from other species with the functional uniqueness. And this principal coordinate analysis is a variance reduction technique that also allows to have Euclidean distance then. And yeah, so this is then a bit of a difference.
And then for a given place in your map, you can play the mean of that for that area, or what? When you put that, when you show those maps in the space, it's a mean for that area, or what is it exactly?
Yeah, so we take the species community in that cell, for instance, and then we take the mean of the risk metrics, so also including the risk measures, weighed by the abundance of the species to include that in some way.
So then you have a value per pixel, I would say, or something like that.
yeah, Brazil or USA.
Okay. Yeah. So, what are you really reflecting with this originality? What is it? Is if I remove a very original
Yeah.
taxa from an ecosystem,
Yeah.
what would happen?
But I know, I think I know what you're
What do you
referring to.
think would happen?
The theory is that if you remove the function of the original species, that affects ecosystem function. And I think you're referring to the scale sensitivity of this, like the comparativeness, which is definitely given. Yeah. So the ideal thing would be to compute this on a much smaller area, potentially by grid cell, but that wasn't possible in the scope of this analysis. And it definitely involves further study as to which extent that will influence it.
So do you think this metric represents or points out the keystone species in a given ecosystem, that if you remove it, the ecosystem can actually change and get to a tipping point that it leads to another state?
Yeah,
Is it that? Or?
yeah, it can. So that is exactly, it depends on the traits that you use. And I actually did a bit of an analysis, or tried to compare it with the literature on keystone species in the net. So what we do, what we well picture here is the trophic networks, and we mostly, the highest axis of variance was body size and K strategists versus R strategists. And these are indeed very important keystone species, so fish have the most influence on ecosystems through their trophic interactions. But it is, of course, not perfect, so yeah.
Can you go to the map where you show the spatial distribution of this function of humanity? Because you say it was buried, yeah. So it was buried on a very small spatial scale, right? Why do you think it's buried on that small scale?
Variance definitely has to do with the sampling coverage. So we have the halls in different scales. We normalize the controlled area, but yeah, it's still, there are some cells that are not sampled, for instance. And then the other, or some areas that aren't sampled. And then the other thing is that we have some outlier values here. So for instance, some schooling species, if they happen to be in the trawling net, that will just boost the abundance in that cell. And that happened in one cell in the Alboran Sea, for instance, that we had, I think, a sardines swarm in them. Yeah.
But on a very high spatial scale, you have probably some spots that are of high functional originality, and just side to side, low points, low location with very low values of that. Why do you think is that?
Well, it depends, for instance, on the habitat heterogeneity. Sorry, the habitat heterogeneity in the area. That is definitely something.
So what would you do to be sure?
Well, it would be super interesting to compute this on smaller scales. For instance, on habitats, the problem is that the data on habitat and substrate is a bit limited in the area.
There is a product of among seawater on habitats, very detailed, 400-meter resolution.
Yeah.
Could be used in a spatial analysis to look at this with temperature and so on. So I think it's a perfect data set for that. And we can go with it.
Yeah.
Thank you. Thank you.
Okay, so first of all, so that congratulations on such a nice presentation and such an amount of work that you did. So congrats on that. You explained really well, and it's a complex topic. You explained really well, so congratulations. I have some general questions. So, have you considered which is the effect of mislabeling, misidentification in your data set? Yes. Have you measured that somehow, or?
No. Short answer, no. The MEDITS data is used by the project and a lot of studies. So there is within that survey just some cleaning and quality measures, but I personally haven't gone into this and that.
Because we know for very related species, it's not the binary misidentification.
Oh, for sure.
Which can say that you're good absence.
Yeah.
The other thing is there any way or what's so the part of you
were able to trade to see interaction among the different species, to measure some level of interaction between the different species. So because I understand the idea of this uniqueness and whatever, but depending on certain interaction that we have between of the species that we are considering, the effect of removal or not, we have further effects. Or is there a way, or have you thought about it?
I have thought about it a lot. I think for now, as this analysis works, it's mostly just possible to, again, depict interactions via the traits. And the analysis, the research background on this also needs further extension. So to really depict all the possible species interactions just via traits is definitely a challenge and relies on a lot of ecological fine-scale studies, which are also spatially dependent and dependent on the environment. Yeah.
Just again, a general comment. What about, because I understand what you're doing is like with certain traits to see the uniqueness, but what about species with quite plastic? What's going to be the uniqueness of that? It's going to get reduced, but on the other hand, it will be quite important to hold certain species that is quite plastic for several of the traits that you are mentioning. What's your opinion on that?
Plastic, as in they have very high values on the traits?
Yeah, so that they could, have some sort of therapeutic practice. Huge principle.
Oh, yeah, okay. Yeah, you're referring to intra-specific variability.
Yeah.
Huge topic in functional diversity, and definitely something that you could go into in a PhD or so. It's a thing, and for example, this also focuses on adult species, whereas larvae are, of course, super important for the ecosystem. So, yes, to make it short, it is important. It's not currently included here because hard to quantify. Well, it's possible to quantify, but with the existing ecological data, it's hard to.
Thank you very much.
Can I just start?
Yeah. Go ahead.
Yeah. Thank you all for being here. I'm Filippo Maria Casarino, and today I will present you my master's thesis project titled Functional Diversity and Seafood Provisioning Under Fishing Pressure and Climate Change, the Barents Sea Cave, which I carried out at the Art University of Melbourne. First of all, functional diversity is the component of biodiversity that influences how an ecosystem works. The functional diversity of a community is determined by the traits present in said community. For instance, the different sizes, shapes, trophic levels, or fecundities present. Seafood provisioning is instead an ecosystem service, which provides humanity with food and job opportunities alike. Seafood provisioning is largely a product of functional diversity. However, the relationship has seldomly been investigated. There is, however, some evidence for a positive relationship between fish functional diversity and fish biomass, at least in some systems, if biomass can be considered a proxy for seafood provisioning. Despite their importance to human life, functional diversity and seafood provisioning suffer pervasive human impacts, especially in the form of climate change and fishing. Climate change can and has altered the distribution of species, hence their functions and biomasses, whereas fishing can act as a selective pressure on traits, for instance, targeting larger organisms or reducing the amount of species that are more of functions that are more sensitive to it. Both these pressures are widespread in the Barents Sea, which is a shallow polar sea found on the continental shelf, covering in between the exclusive economic zones of Russia and Norway, with a small area of international waters in the middle. The main oceanographic features of the Barents Sea are three: the inflow of cold Arctic water from the north and east, the inflow of warm Atlantic water from the southwest, and the polar front, a central area where these two first features converge, which serves as a divide between boreal and Arctic communities. Arctic fish communities are dominated by small, specialist bentivores, whereas boreal fish communities are dominated by large piscivores generalists. Studies have found that Arctic communities have generally lower biomasses and lower functional diversity compared to the boreal ones. The Barents Sea is among the fastest warming areas on the planet. This warming has led to the so-called borealization of Arctic areas, whereby Arctic species are replaced by boreal ones. And it also has a long history of overexploitation, which has caused several stock declines. This study only focused on the western side of the Barents Sea, covering the Norwegian exclusive economic zone and in international waters found at the center due to data availability. The aim of the study was to, for the first time, jointly address the relationships between anthropogenic pressures, functional diversity, and seafood provisioning, as well as the relationship between diversity and provisioning. And based on literature, I expected the following relationships. I expected warming to positively influence provisioning and diversity, whereas I expected fishing to negatively affect both provisioning and diversity. And lastly, I expected diversity to positively influence provisioning. So, to answer my questions, I collected the environmental data on fishing activities, functional diversity, and seafood provisioning. As measures of warming, I collected the mean chlorophyll A concentration, as well as the mean and standard deviation of sea ice concentration, sea surface temperature, and sea bottom temperature. In this case, the standard deviation is meant to be a measure of the seasonality. As a measure of fishing, I recovered data on total fishing effort. Whereas for functional diversity, I used bottom troll survey data, as well as trade data of fish species of the Northeast Atlantic to compute functional diversity metrics. Functional diversity can be better studied when decomposed into its three main dimensions at a community level, which are functional richness, the number of functions present in the community, functional evenness, the regularity, how regularly spaced they are, and dispersion, a measure of their spread, how different they are from each other on average. I later used the same bottom troll survey data to compute the total fish biomass as a proxy for seafood provisioning. All the data I used covered the time range from 2004 to 2022, except the fishing data, which started from 2012. Then, to better understand my variables and the study area, I mapped both the mean values of each variable and their slopes, their annual rates of change, to visualize spatial temporal patterns. And later I focused on the relationships between the various variables using spatial temporal generalized linear mixed models. Moving to the results, the environmental variables pointed at the presence of unexpected thermal gradient in between the south and the north, here exemplified by the abundance of sea ice in northern and northeastern areas, whereas their annual rates of change highlighted the presence of the evidenced warming, here represented by the decrease of sea ice, where it was most abundant. Fishing was instead widespread, with some hotspots along the continental shelf and along the Norwegian coast, and it varied less, but generally it had some hotspots of increase in the south, where it was already quite high. As for the diversity metrics, they all displayed very similar spatial and temporal patterns. So here I exemplify them all using just functional richness. They all displayed higher values diagonally across the study area, somewhat matching the position of the polar front. And as for their annual rates of change, they displayed increases, especially in the north and east. Fish biomass, was instead higher in the southwest, and it increased especially in the north and the center. Moving to the relationships, among the available covariates, the only one that I found to be supported in models as a driver of functional richness was sea ice concentration with a negative effect. Whereas functional evenness was positively driven both by mean sea bottom temperature, mean chlorophyll A, and the seasonality of sea bottom temperature. Functional dispersion instead, the functional dispersion model instead did not support any covariate, so I'm not displaying it here, and notably no functional diversity metric was related to fishing. The biomass model instead supported warming indicators as positive drivers, fishing as a positive driver, and functional diversity metrics as negative drivers. So particularly sea bottom temperature and its seasonality, as well as sea surface temperature seasonality, had a positive effect on biomass. Fishing had a positive effect on biomass, and functional richness and evenness had a negative one. So out of these expected relationships, I observed these ones. And particularly, there are three unexpected ones. The undetected relationship between fishing and diversity, which I expected to be negative. The positive one between fishing and seafood provisioning, which I expected to be negative and I found to be positive. And lastly, the negative one between functional diversity and seafood provisioning, which I expected to be positive, but I found to be negative. Digging deeper into the possible explanations to these relationships, the positive one between diversity and warming could be explained by the presence of environmental filters. For instance, sea ice was a negative driver of functional richness. Sea ice imposes very strict physical limitations on its associated communities. For instance, it limits light availability, and of course, it is associated to lower temperatures. So this could limit the number of functions that can persist in ice-associated habitats. As for the positive relationship between biomass and warming, it can very well be explained by the presence of larger species in boreal warm water assemblages. Whereas the undetected relationship between diversity and fishing could be explained by the long-lasting presence of this pressure in the Barents Sea. Indeed, the first industrial trawling activities developed in the 1920s, and they've been steadily present ever since. So it is possible that the most sensitive species, or better functions, have already been affected. Hence, the community might have already restructured in association with fishing. And then the positive relationship between biomass and fishing is most likely not causal. I interpreted it as evidence of the active pursuit of fishing vessels for more productive fishing grounds. And lastly, the negative relationship between biomass and diversity, which was unexpected, could be explained by species dominance. Indeed, one species, Atlantic cod, Gadus morua, dominated the survey by biomass. This is a generalist piscivorous species, so its high abundance in certain areas could limit the presence of other functions, for instance, due to predation. Indeed, this species is often involved in top-down control mechanisms in trophic chains. The high abundance of cod in some areas could also lead to the over-representation of the functions cod bring to the community, hence reducing regularity, hence reducing functional evenness. Thus, these two processes explaining the negative relationship between biomass and both functional richness and evenness. Another explanation I found was that mixing could explain the higher levels of functional diversity, here again exemplified by functional richness, in the area somewhat close to the polar front, as here the two zoogeographical areas merge, the boreal one and the Arctic one, hence explaining by mixing the higher number of functions in this case. This area was also surprisingly characterized by lower biomasses, which cannot be explained, for instance, by the thermal gradient, and may indicate that in these in-between conditions, oceanographically I mean, there may not develop high biomasses. So the take-home messages of my study were that diversity and provisioning increased with warming, that the expected negative effect of fishing remained undetected, and lastly, that biomass was higher where diversity was lower. And I would like to thank all of those who contributed to the project, all of you for listening, and for your attention. I'm ready to answer your questions.
Great presentation. I really enjoyed it. Very nice frame. Very nice presented. It reflects the amount of work that you have to do. Yeah, I'm going to talk about I want a little bit more detail on some of your goals.
Oh, yes.
Yeah. But especially on because of you. You did like series analysis, right?
It's kind of, yeah.
Per pixel, you analyze the.
You want to know the resolution,
Exactly.
the spatial temporal resolution, yes.
Yeah.
So the most limiting variable was the community, like the sea bottom troll survey, the bottom troll survey data,
Yeah.
which was at a whole level. There's a surveys carried out every year in a somewhat regularly spaced grid of stations 65 kilometers apart. And so that was the limiting variable. The functional diversity matrix and the dynas were the limiting variables. If you want, I can show you. Yeah, so these were the sampling spots for the study area. And yeah, so the other variables, I could get a raster of the whole area.
Yeah.
So then I extracted the raster values at each hole location. So this is all hole based. Every like a sampling position in space and time. Once a year. Once a year in. Oh, wait. The biological data is once a year. Temperature I had for the whole year from Copernicus.
Every day.
No, I downloaded it every month.
Every month?
It was very long computation. So that's why I have mean value of the year and its standard deviation as a proxy for seasonality. And then I extracted the values at each whole location.
So you calculate a mean of the year of the temperature. For
Yes,
each pixel.
for
And
each position of
then
the whole.
calculate the linear trend for that.
Then, no, not for each position. Then I used like the random term of the mixed effect model to account for spatial and temporal correlations. And the positions of the whole are not, the positions are not the same over the years.
But if you have a good spatial resolution, the power resolution, the best thing is to take advantage of that.
Yeah, I can.
And calculate the trend with all the data you have. So you can do counts, for example, to remove seasonality from the data and get the linear trend with removing the seasonality. Because in this analysis, the seasonality is mixed with the long linear trends that might be or might not be there. You know what I mean?
Yeah. So like you mean temporal autocorrelation?
Yeah.
Yeah. So I try to address that to like using an AR1 spatial for autocorrelation structure. So that should account for it.
Okay.
Or it does its best. Because I was thinking about doing like a time series analysis, but you needed the same position over time, and so here they change. So
Yeah, but for the environmental data, you have the same position over time.
yeah,
All
for the environmental one.
the same pixel.
Yeah.
And about the conclusions. According to your conclusions, would it be a good idea to take diversity as the main measurement according to what you should do conservation policies? Because it increases biodiversity. Increases.
Yeah, it increases, and
So it's good.
that is no.
So
I mean, changes in biodiversity are not necessarily good because they are positive. And of course, the Arctic community is being replaced. So what we expect is that in the long term, this mixing in intermediate areas will actually turn into like simply replacement of Arctic species. So the Arctic species are already stuck at the top of the range and they might disappear. Especially because the continental shelf does not progress much longer than there is the deep Arctic Ocean.
okay, thank you very much for the presentation. Thank you, Ingrid.
Yeah, so congrats on the presentation. Clean presentation.
Thank you.
Easy to follow you, even though this is not my area of expertise at all. But well done. I think you did a great job guiding us to the topic. So well done. So I have I really like this kind of study. I think they're really necessary. This kind of study. But I always ask my friend, we have some sort of internal control that things that you know that due to the data that you are handling, you should pick up. You know what I mean? That some like data or partial data or something that you should be able to detect. certain parts that are known that we're detecting, like internal control for your data, you know what I mean?
You mean like
Yeah,
expected from literature, like
Yeah.
or biases in the data?
No, expected from it. Something that you know that you
Yeah.
should, by analyzing that, which is certain obvious thing
Yeah.
that you need to pick up and say, okay, you know, I need to pick it up.
Yeah, I'm doing it right.
Yeah, like internal control that to say,
Yeah.
okay, this is processing in the way.
Yeah.
Something, you have something like that in the data?
Yeah, so this is an interesting question because I'm using, like, the Barents Sea has been investigated for its functional diversity, but it's been through the years and methodologies have been updated. So I used a methodology that had never been used in the Barents Sea. So I used previous studies as comparisons, but at least the main patterns I was able to find. For instance, higher diversity, somewhat close to the polar front in the central area. And then this was also, I mean, at the start it was harder, but then I became better at it. But at this, like, a lot of my colleagues said that my patterns look weird, but actually this is because most studies, all studies, focus on the whole Barents Sea. But I couldn't have access to that data. So my patterns looked weird, but because everyone was used to see like the whole overall pattern, but then looking better into it, it makes sense. And I think it develops on some of the previous studies. For instance, some studies back from 2013 predicted that richness would increase in Arctic areas, and this is what I somewhat found.
So the other thing that I'm sure you will discuss, again, this is not my area of expertise, but what about because we are using this environmental data, how many, so are you considering also for this diversity species that somehow part of the life history trait or life history are not in that way? You know what I'm saying?
Yeah, I know. And this is another.
So, but maybe some of the species that you're analyzing were not there, but critical phases of the life are not from that particular, and how that provide or give some noise to the system. I don't know, maybe it's too
Yeah, no,
big,
this is a very good question, and it's very relevant for the Barents Sea. Indeed, as I told you, cod was the most abundant species, and cod is a migratory species, at least the large pelagic stock, the one that's like the largest on Earth. And they all come to spawn in this area, which is out of the sampling for me, but they reproduce in a different season. So when the survey is collected, cod is back into its main year habitat, not where it reproduces. But of course, there could be other things going on here that affect then the recruitment of the species. But that increases the complexity of the study gigantically. So yeah, and this is also true, for example, for haddock, the second most abundant species, and for many others. But generally, not for the Arctic species. So that's
Okay, once again, congrats.
Thank you very much.
Good job.
Thank you, Ron.
So, really, really nice. I appreciate it also that you didn't put a lot of writings in the presentation. Can you go back to the models?
Yep. The model outputs. A specific one? Or a specific one?
Yeah.
Any?
Generally.
Yeah. Okay, that's most complex.
So the estimate is giving you how the two are correlated, no?
Yeah.
Okay. They are not super high.
No.
Okay, so you think that maybe this the factor that you consider that explain just, or there are other factors that explain better the situation.
Most likely,
Yeah.
but
You have
I think,
an idea of which
yeah,
ones?
yeah. I think it is a very complex system, but I believe that just these variables, if recalculated in a different way, could bring some additional results and explain maybe more of the variation that we see. For instance, in the case of fishing, exploring how fishing affects communities on longer scales rather than in the year. And the same is true for temperature. I know, for example, that heat waves have caused dramatic shifts of communities, such as Arctic communities, but then after the heat waves, the Arctic communities are not so motile, so they take time to come back. So you don't pick, in my model cannot address for that. So it still finds an area that is cold and doesn't have an Arctic community, but that is because it hasn't come back, if it ever will, of course. So yeah, there are a bunch of other variables. I had a lot in this study. Now you see only a few that were selected.
And just another thing, then I don't know if it was possible, but maybe could adapt to merge the two together, the fishing pressure and the same amount of fishing pressure and the diversity.
Yeah, to visualize.
Yeah, to visualize better. So for someone that is listening to you, it was easiest to catch, you know?
Yeah, I know, but
Yeah,
I don't
no,
think
it
like
was just
I think like the very big like the plots that show something going on like that show some correlation are mainly the temperature ones, the warming ones,
yeah,
and
yeah,
the diversity. But for fishing it was.
Yeah,
Yes.
fishing, but I did fishing, but just because I was studying one with other factors.
Yeah. Thank you very much.
Thank you so much. We have some time, so anyone from private want to make questions?
Yes. Can you go to your expectations as well, like the interactions between the different
Yep.
aspects?
Sorry, overshot.
Yes. So, if I remember correctly, you said that you expected that fishing would negatively affect provisioning, right? And then you found that it affected it. Why? Maybe I don't understand why did you expect that more fishing would mean effectively less provisioning?
Yeah. In this case, for provisioning, I've used total fish biomass. So I expect that because fishing directly extracts biomass, I expect future biomass to be lower, or my biomass at the same time to be lower. So that's
provisioning did not mean like the
seafood
delivery of seafood.
Yeah. That's what I mean. That to capture it, I use total fish biomass. So the total fish biomass available in a specific time. And then I try to understand if fishing was a driver for that. So if more fishing means, let's say, more biomass depleted this, and so the opposite. But that's not what I found. But
What could be an alternative instead of biomass? What other parameters maybe you can use to?
like what? Like how fishing can affect?
Yeah, to estimate.
Oh, what instead of?
Yeah.
This is a tricky question.
Yeah.
I mean, the good thing about biomass in this case is that it is spatio-temporally explicit. So I know where it is at what time. Whereas other indicators that I could have used, maybe land, maybe I don't know, future landings or something like that.
That there you can have some bias because the biomass data that happens because the vessels are just going where the fish are. So it's some kind of bias in the biomass data.
No, the biomass, I don't think the biomass data is biased. It's different data sets. Like fishing, this is fishing independent biomass data. So the survey is scientific and doesn't have to do with fishing. Yeah. So I'm not sure. I thought this would be the best indicator for seafood provision. There are others. You could look into the output, how much biomass is landed, for instance. So maybe more fishing in the end leads to less biomass landed because of erosion of the biomass available. But that would be really for the whole area. Like I can get that data, but it's like for the whole area, so it loses the.
How you would explain for fishermen, for example, that in this case farming is good for them.
Yeah.
More biomass, more provisioning. So.
Yeah. That is what could be expected from this. That the biomass will increase, and overall, the main stocks in this system are cod and haddock. They will increase these two species, but it's not that simple, of course. There are many other things at play, and this doesn't necessarily mean they won't.
Maybe what is good, just the local leaders is not good. I mean, it shouldn't.
Yeah.
Maybe at some points you can have a benefit,
but also going to this map. Here you can see that in the southwestern area, biomass was increased a bit. So maybe this is a hint that, well, yes, biomass will increase further up. In the end, it will decrease below. So the whole thing is shifting. But this, of course, I cannot say.
Okay. Congrats,
Thank you very much.
Okay. Last but not least, presentation is from Francesco.
Good morning, everyone. My name is Francesca, and I'm very happy today to present my thesis, which focused on benchmarking the use of eDNA metabarcoding for biodiversity assessment in the marine protected area of the Tremiti archipelago. My supervisors are Giovanni Ghimenti and Dr. Larissa Frue and Dr. Giovanni Ghimenti. Environmental DNA can be defined as any DNA, intracellular and extracellular, found and recovered from an environmental sample. In my case, it was sediments, but it can be water, ice, or any environmental matrix. It is becoming more and more used in science because it's not invasive, it's cost-effective, and it's a very useful tool for the early detection of alien species. In the Mediterranean Sea, which is one of the most studied marine regions in the world, previous studies have been conducted with the use of eDNA, particularly to spot the Monk Sea, which is not easy to spot with traditional methods, but also to study phytoplankton and benches. The Tremiti Island MPA is located in the South Adriatic Sea, so we are more or less in the Apulia region. It is made of 5 islands, 3 main bigger ones and 2 smaller. Pianosa is 12 miles north from the others. It's a marine protected area since 1989. It is part of Natura 2000 because it's an important area for birds, and also it presents many habitats that are of high conservation importance. My study is the first one using the eDNA in this context. The aims of my thesis are through eDNA metabarcoding of the cytochrome oxidase subunit 1 gene starting from sediments to characterize the community composition across different habitats, explore patterns of beta and alpha diversity, and evaluate the strengths and limitations of using eDNA in this context. The research questions are: how does metazoan community composition as detected through eDNA metabarcoding vary across different habitats within the Tremiti Island NPA? And how effective is the eDNA as a tool for biodiversity assessment in this Mediterranean NPA context? The habitat types that were sampled are these 6. We have Posidonia oceanica meadow, also known as Neptune grass. It is an endemic marine plant of the Mediterranean, and it's a well-known hotspot of biodiversity and nursery, shelter, and feeding area for many species. Coral forest, I sampled mainly coral forests made of Emicella cavolini and Paramuricia clavata. It is also this habitat known to be a shelter and a nursery for many species. It is the deepest habitat that I sampled. Cladocora caspitosa pet. Cladocora is a colonial coral endemic as well of the Mediterranean, and it is also a quite rare habitat in the Mediterranean. Sand, which is a more open and dynamic habitat with poor three-dimensionality, and in the end, submerged and semi-submerged caves. The first are fully below the sea level, the latter are partially below the sea level, and the main difference is that semi-submerged caves are way more dynamic habitats than submerged caves. This is a map of the MPA. Pianosa is not in the map because we didn't have the permits to sample there since it's the no-take zone. So all the sampling happened across these islands in June 2024. As said before, I sampled the sediments. In this picture, you can see that I just scooped the sediment from the bottom. I sampled 2-3 dive sites per habitat based on the extension of the habitat, and 3 replica per dive site were collected. The DNA extraction happened in Bari. I used the fast DNA kit from MP Biomedicals. These are the primary use for the amplification during the PCR. Both the PCR and the sequencing were carried out by an external facility in Bari, but I had the chance to follow it. For the sequences and the data processing, I used the data2 pipeline and the function assigned taxonomy and BLAST to compare my sequences with the known databases. The data analysis was carried out on RStudio. I calculated the alpha diversity with 3 different indices. I ran a permanova on that, and then I calculated the beta diversity, and I ran a permanova to check if the community composition across habitat was significantly different. In the end, I did some taxonomy plots. Going to the results, not all the sample collected amplified. We have here in this table the mean DNA concentration after extraction. So we can see that even if the concentration was good for the amplification, not all of them amplify. So these are the numbers of samples per habitat that in the end were sequenced and used for the analysis. Looking at the alpha diversity, as said before, I calculated richness, Shannon, and Simpson. Richness taking account only the number of unique sequences, while Shannon and Simpson also assess for the evenness of the species in the habitat. Only Shannon came out significant, but when running the Tukey test, it didn't show any significance in the pairwise comparison among habitats. What we can see from here is that all the box plot more or less overlap. The highest median in richness is from Posidonia oceanica meadow, while in Shannon and Simpson it's in sand. The lowest median across all of them is found in submerged caves. We were expecting a high diversity, of course, in Posidonia oceanica meadows, but what surprised us was the high diversity found in sand, because as said, it's a full three-dimensionality habitat. So the possible reason for this data is, first of all, the presence of infauna and borofauna present in the sediment, which in this study is actually biased towards, since I sampled the sediment. Another possible explanation is the presence of larval DNA that from the water column just settled on the bottom and came in our samples. Looking at the beta diversity, here we have an MNDS plot. We can see that each polygon is a different habitat. We can see that they don't overlap. The Permanova was actually significant, so we can say that the community composition in the habitat differed. What was interesting is that we noticed this clustering. So in the orange circle, we can see all the euphotic habitats, so Sen, Posidonia, and Cladocora, while in the blue circle, we can see all the mesophotic and aphotic, in case of the submerged caves, habitat. This tells us that the light influence the community structure, and also we expected, and this aligned with our expectation, that the two types of caves have a more similar composition, since the fauna there is probably more specialized for that type of habitat and that type of conditions. Looking at the taxonomy, here we have on the x-axis the habitat, on the y-axis the abundance. In the first two graphs, the abundance is just the number of breeds, while here I transformed that in percentage to address the difference of samples number, but also to better appreciate the community composition. On the left, we have also the presence of unassigned sequences, and on the right, I took them out again to better look at the composition. We can see that in habitats such as Posidonia, Cladocora, and submerged caves, the presence, the number of unassigned sequences, which is the dark blue, it's very high, because here they drop almost drastically. We can see in light green that Annelida is the most abundant phylum across all the habitats. The orange are Arthropoda, which are also present across all the habitat. In red, we can see Porifera that are in semi-submerged caves, which makes sense. Maybe I forgot to mention that the color code represents the phyla present in the samples. So, in particular, the vast presence of Annelida aligned with literature, since Annelida is the third most abundant phylum in the Mediterranean, and 6.6% of the species in the Med seems to be part of this phylum. In this plot, I included the phylum with the color coding, but I also added some class to better look at the diversity in this habitat. The sides of the circle represent the abundance in sequences. We can see here that among the Annelida, the Polychaeta are the most abundant. The Posidonia oceanica meadow has the highest number of classes and is followed by scent. Also, Malacostraca and Demosponge, Malacostraca here and Demosponge here, are present across all the habitat, like also other phylum. In particular, Malacostraca, the only two species we found are two alien species. The first one is Calinectes sapidus, also known as blue crab. It is from the Atlantic, but is well established, sadly, in the Mediterranean. This is the first ever evidence of this crab in the Tremiti Island. Ecologists thought that the Tremiti Island were safe because the conditions there are not the best for this species, but we actually found multiple ASV assigned sequences to this species with a confidence higher than 99%. Moreover, we had the highest peak count of sequences at almost 60,000 from a semi-submerged cave, so we are quite confident that the DNA is there. The other species is this Panopeucai. It is also from the Atlantic and it's established in the Med only in Sardinia in Spain. This is the first record in the Adriatic, but even if the confidence was high 100%, the reads were around 700 and only in one type of habitat. So better research must be done to be sure that this species is actually there. In the end, I encountered many limitations. The lack of amplification, especially in some samples from the caves. This might be due to the fact that organic compounds inhibit the amplification of some sequences, or also the fact that the region that I targeted was actually not there in the samples. We had a lot of unassigned SVs, as we saw. Only the 14% of 3,000 unique sequences were assigned to class level. And this is also a well-known problem in the data when using databases. From my samples, I only had Benthus species, and we didn't find the habitat former molecular trace. So this also highlights the need of a better resolution in databases. And in the end, I also detect some taxa that are known to be not present in the Med. So it was probably a mistake in the database. So we can go to the improvements, and so what we would do better would use multiple primers, so not only one, but more than one, targeting different also type of organisms. Samples also the water and not only the sediments. And in the end, would be very nice if there was a Mediterranean database with the sequences of all the species present there. In conclusion, we can say that the Benthic community composition varies among the habitats in the MPA, that the eDNA is confirmed to be a reliable tool for the early detection of alien species, that the interpretation of biodiversity pattern in dynamic habitats like SEN is tricky, and that there are big gaps in the reference database that need to be filled. So to wrap up, I would say that the use of eDNA for biodiversity assessment is a reliable tool, but in its improvement to be successfully used in the Mediterranean. I would like to thank you for your attention, and I would like to thank my supervisors for all the help that they gave me. Thank you.
Yeah, 34 for congratulations. I really enjoyed it. I did have the pleasure to read the manuscript, but everything I did not. But everything was super clear. I really like the way that you explained how you guided us through, and also how you acknowledge the limitation. I told the previous one. I think this is really important to acknowledge it. So I think the piece of work is really important. It's key to providing the first evidence for many things. So it's the first time that this kind of study was done in that region. It's super important. The first step. But to acknowledge some of the limitations, I think it makes your work even more progress somehow. Yeah.
Thank you.
So well done. So it was funny because I was before you presented the result, I was on a different type of habitat, then different potential inhibitors to amplificate. In fact, you find something like that.
Yeah.
I mean, at least in coral forest, you have out of 9 examples that you choose only 5 of them amplified.
Yes.
Have you tested if there was some inhibitors there?
No, we didn't test for any type of inhibitors in the samples because we didn't have time. But they would have been very interesting, and also if we had the chance, it would have been important because we could have process the samples in a different way, clean them, and then try to amplificate them again to then do the sequencing. So sadly, no, but would have been nice.
So, and of course, this is the first stone that you are, so it's the first step, but I think more about temporalis, seasonality of your data. So, what do you expect that it's going to happen if you go instead of June, go in December? What do you expect?
So the study was carried out in June. I would say that since the Mediterranean doesn't have like big change of temperatures over season, we don't have big changes on physical characteristics of the water. I would not expect a big diversity among the different seasons, honestly. But that would also be an interesting thing to test for in a future study.
That's one thing that I mean, maybe we cannot do everything in one single study, of course, and we acknowledge the fact with the primary bias you might have using cocktails that might grow your assignment and so on, and the relative representation of your sample. And what about, because of the series, I think it's feasible maybe to find similar habitat but close by but not really within the marine protected area to see how different.
So that's an interesting question.
I don't know
we discussed about this with my supervisors at the start because, as you can see, we don't have any negative control. But that's because this habitat can be found in the Trinity NPA and not outside. So no, they are in the Med, but they are not like immediately out of the NPA, so you cannot compare if the protection is actually working in that area or not. We could take samples from the same habitats in other places in the Mediterranean or other places on the Adriatic coast. But yeah, there was a discussion at the start.
But don't you have any of those 5 habitats that you were something close by at least to have at least a request for one of those habitats?
No, we don't.
Is it close to one that we have?
No, we don't. We only sampled inside the Trinity. Yeah.
Okay, great.
Thanks.
Thank you, Ron.
The presentation was really nice, actually, as well. Just one thing, ecologically speaking, the Mediterranean has a high variation in the ecological habitat of the water. I mean, if you think just maybe in the Trinity Island it's less highlighted, but if you think at here, the Adriatic Sea, we have 30 degrees in the water in the summer and low to 5 degrees in winter. So we have specific organs that are able to survive, and also seasonally, if you think of algae, hydroids, so there are many organs, taxonomically, different taxonomic levels that have a high seasonality. So maybe you couldn't find changes. I don't know that if this could be detected because I don't know about the sequences that are present online, but I'm sure that some differences could have been detected. Do you have regarding first of all, the sequencing allows you to differentiate at phylum or class level, or also at species level?
No, I was able to arrive at the species level, but the total amount of species were around 400, so it wasn't possible to show it in any graph. I would have loved it, but
the analysis were conducted at species or species level or on a higher taxonomic level?
It depends on the analysis. So the alpha and the beta diversity were conducted on the ASV actually, so not on the species level, not even on the phylum, just on the different ASVs found. While the plots, the taxonomic plots, were conducted at a phylum level and the class level, the second one.
Okay, and the PCA or NMDS, I don't remember.
The NMDS, yeah, that is conducted on the unique sequences found before the assigning that sequencing.
Okay, so
species.
regarding the submerged, the completely submerged cave, at which depth were
The Samnet caves, I think they were around 18 meters, 17 meters. Yes.
Yes, because depending on which level you take the analysis, it's shown that maybe they have a very similar composition, but I mean, if you consider, say, some batch caves that are at the sea level or at the 30 meters, I mean, some differences have been found because they can host very different species. So they consider only the current, as they said, the development dynamic, no? So, and just another thing, considering that, so the sand was the only habitat that you collected, actually the sand and not the water.
I never collect the water.
Ah, okay, so
it's
how
all...
did you collect it in the coral forest?
Well, that's a good point, because in the coral forest, as you can imagine, as you know, so coral forests are mainly on a rocky bottom substrate. So I collected the sediment like at the bottom of the coral forest, or where I could find any like small amounts of sand. I was like,
so you didn't collect it in water?
No.
Okay.
And that's probably the reason why all the species that I found are mainly benthic.
See, that. And you had a sort of a ground truthing of the species that you found listed in your name. So there are species lists available that help you to say, okay, what I found is actually there.
Yeah. So since it's an MPA, there's monitoring going on every year, and my supervisor is the one doing it. So we were able to compare the species and say, okay, this is actually there. And actually, the one listed here are the species that we found not present there. So yes.
So it was based on the geography.
Yes. Absolutely.
Okay. Thank you.
Congratulations for events. They were very nice, very well framed, very easy to follow. I also like a lot that you admit the limitation. This is very important. It's actually not my field, but I have a very easy question. And what about the scale of dispersal of this EDNA in the Mediterranean? For how long can it stay in the water column to travel from one place to another due to affection by currents?
Yeah.
Do you have any?
Yeah, so in literature, it really depends on the condition, even of the day, water temperature, light penetration, salinity, everything has an impact on the DNA. In literature, it's reported that DNA can stand in the environment up to 2 days, 48 hours even, but it really depends on the condition of the moment. For example, I did also my professional practice with eDNA, and we were supposed to go sampling, but since it was raining a lot, we couldn't sample because the introduction of fresh water in the sea was a problem to collect the DNA. So, yeah, that's one of the biggest challenges of eDNA, actually.
Also, one observation I agree with you that to conduct a seasonal study would be very difficult. Extremely seasonal seasonality, temperature, and primary conductivity here is the highest all over the northeastern Atlantic. So, yeah, seasonality is important. And I wanted to ask a question about the MPA itself.
Yeah.
Is it really an MPA or is only an MPA on paper? Because this happens always.
Well,
everybody knows.
We are live streaming, so I don't know if I can answer. There's some reinforcement in the zone B. There's a boy camp, no back camp. There's a boy camp, I think, to avoid the anchoring of boats. But sadly, it's a very well crowded area in summer. and I think it's very hard to reinforce the protection there. So I don't know. I don't know. I don't know. Yes.
Okay, thank you very much.
Thank you.
Thank you so much to Francesca. Let me say congrats to all the students. You cannot imagine how proud we are because many of you saw me the first semester and it's difficult to imagine that many of you at the end, after 2 years, remain so great presentation. So congrats, enjoy, and the best for all of you. Okay? So congratulations, Francesca. Goodbye, see you in the graduation. Bye.